Magic sir gone. Yes, you can see me now, right? Amazing. Let's not waste any time and I'll start with the key takeaways today. The key takeaway today is going to be the entire syllabus. Today we'll be covering the entire syllabus. It might take 3 hours. Just a disclaimer to everyone, just a disclaimer, this session is going to be really long because I want to explain each and every line of NCERT. I want you to understand each and every word of NCERT, each and every concept of this chapter. Right? For that, it is going to take time. So let's do it. Okay. So the takeaways. What are the key takeaways from today's class? Right? What are the key takeaways? What is the flow? First, we'll be starting with what is blood and what is lymph. First topic which we're going to study is what is blood and what is lymph. What is blood made up of? That is, then we'll be talking about coagulation of blood, what is blood grouping, that is, and we'll be learning about erythroblastosis fetalis, then heart diseases, then we'll be learning about circulatory system, different types of circulatory system, right? Then we'll be learning about human circulatory system, human heart, the most important part here, the human heart, the blood vessels and the type of circulation. In the end, what is cardiac input, what is cardiac cycle. So many topics today, so many concepts we'll be exploring today. Okay, then what is nodal tissue, what is ECG, that is electrocardiogram, and regulation of cardiac activities with the help of hormones, how different hormones, how medulla oblongata, the brain, how brain is involved in the blood circulation, how brain plays an important role in the circulation of heartbeat. Amazing. Sir, we are 45 minutes late. Exactly. Oh, no, the session was 7:30 because I changed to 7:30, but we are 45 minutes late and we will go. Let's start with the first one. So, when I asked you the first term, when I asked you a question before, which cell in the human body? Ready, students? Ready? Let me know in the ready if you're ready. I am very afraid, Kiran. Please guide me. Sir, we are 45 minutes. Okay. Students, if you're able, if you're ready, thumbs up in the chat. Thumbs up in the chat if I can start. Thumbs up in the chat, all of you students, I want thumbs up in the chat, and I'll start with the first topic, that's going to be blood. Thumbs up in the chat, each and every one of you, thumbs up in the chat, and we'll start with the blood. Let's start blood. So what do you mean by blood? Is blood a connective tissue? First simple question, is blood a connective tissue? Yes sir, blood is a connective tissue. So what exactly is a blood? Recall, blood is a specialized connective tissue. Blood is a specialized, amazing connective tissue. Without blood, we cannot even imagine our life without blood. That's how important blood is. That is, it is a fluid matrix. Blood is a fluid. It's fluid, it is not solid in nature. Blood is fluid. That is, it is connective tissue. That is, it links and supports other tissues. Right? Blood is a connective tissue which supports and links other tissues, and it is also has cells and matrix. It has cells and it has matrix. That is the blood. Blood is a connective tissue which is, exactly, blood is a connective tissue. So what is the function of the blood? If I ask you, why do we need blood in the human body? What is the need of blood? Right? The main need of blood is what? Delivering the oxygen and CO2. We know, we have been learning from younger classes. Always, sir, blood helps in transportation, right? Blood helps in transportation of what? Oxygen with the help of hemoglobin. That's the basic concept all of us have been learning from a very young age, right? From very young age, we've been learning the blood is a connective tissue which helps in transportation, of oxygen and CO2. It carries nutrients, waste products and hormones. It also carries what? Hormones. It helps in maintaining the pH. It helps in maintaining the pH temperature. Right? Blood becomes hot. Right? Helps in maintaining the pH, helps in maintaining the temperature. Clot formation. Students, who can tell me in the chat which is the part of the blood? Which part of the blood helps in clot formation? I'm looking in the chat right now. I want to know the answer for which part of blood, which component of the blood helps in clot formation. Let me know in the chat. Let me know in the chat which part of the blood. Which part of the blood helps in clotting and also helps in defending against pathogenic organisms? That is, blood also has what? Clotting effect, and also the antibodies are present in the blood. Exactly, the blood platelets. The blood platelets helps in clot formation. Blood platelets helps in the clot formation. Amazing. Thrombocytes. Bala is saying thrombocytes. Is thrombocytes correct answer, students? Let me know in the chat, is thrombocytes other name for platelets? If yes, thumbs up in the chat. If yes, thrombocytes. Exactly, thrombocytes. So where do these blood cells originate? What is the origin? Where do bloods originate? Students, a question for all of you. Where do the bloods originate? Let's see who can answer. Thromboplastin, right? Thromboplastin, it's thromboplastin, right? Ravindra Nani, it's thromboplastin, which helps in what? Activation of thrombokinase. Thrombokinase converts, right? Thrombokinase helps in fibrogen to fibronin. We'll come to clotting, don't worry. We'll come to so we'll come to that. So people are saying the origin is red bone marrow. But where do we find this bone marrow? Students, where in which part of the bone do we find bone marrow? Yes, bone marrow, red bone marrow. But where is red bone marrow present? Who can tell me in the chat, where do we find the red bone marrow? We know it is red bone marrow, but where in the body? Where do you find the bone marrow? Where do you find the bone marrow? Who can tell me? Bone marrow is found in your long bones, such as which is the longest bone in the human body? Which is the longest bone in the human body, students? Yes, also in liver. We'll get to that liver part. We'll get to the liver part. We'll get to the liver part, but right now, which is the longest bone in the human body? Students, in the bones, at the end of each bone. Here, for example, that is, okay, we'll get to that red bone marrow, but there is one more term which you need to know is hematopoiesis is the meaning of, is the formation of blood, is nothing but blood cells. Poiesis means what? Poiesis means to make. So the making of blood has a name called as hematopoiesis. That is, formation of blood cells is a process that is by which blood cells components are formed. So hematopoiesis is a formation of blood. Femur, exactly. Femur is the longest bone. So can you tell this is your femur? This is your femur. Towards the end of the femur, right? The spongy bone containing red marrow, the ends of the femur. Can you see this is the end part? The end part has the spongy part, and this spongy part has what? Contains the bone marrow. This is your complete bone which has blood vessels and bone marrow, but towards the end region of a bone we have the spongy bone, and in this spongy bone you can find the red bone marrows. Amazing. Sir, can you keep your mic more near to you, please? Students, you're not able to hear me? Students, you're not able to hear me. The mic is almost near my mouth. Dog biting bone, exactly. Students, are you not able to hear me? Check. Students, are you not able to hear me? Increase the volume. Increase the volume on your phone or laptop or computer or tablet. Because this is the closest I can. Next thing I have to do is keep the mic inside my mouth. So I cannot do that. So I told you what is hematopoiesis. Hematopoiesis means what? Formation of blood. Right? Formation of blood is hematopoiesis, but where does it happen? In the spongy bone, which contains the red bone marrow towards the end region. Clear? Next slide. That is hematopoiesis. All blood cells arise from hematopoietic stem cells. So we have something called as hematopoietic stem cells, and these stem cells will give rise to all the components of the blood. That is your erythrocytes. Students, who can tell me what is an erythrocytes? Is erythrocytes RBC? Is erythrocytes WBC? Let me know in the chat. Is erythrocytes RBC, red blood cells, or white blood cells? Erythrocytes, leukocytes, and thrombocytes. Someone was telling me, Bala was telling me, thrombocytes are the platelets. These are the platelets. Thrombocytes are the platelets, and we have erythrocytes, which are your red blood cells, and leukocytes, which are your white blood cells. Clear? Now, first one is erythropoiesis. Erythro means what? Erythro means red blood cells. Poiesis means to make. To making of red blood cells is called as erythropoiesis. That is stem cells, hematopoiesis, erythrocytes. Erythrocytes, formation of erythrocytes is known as erythropoiesis. Now what about leukopoiesis? Leuko means what? Meaning of leuko is white blood cells. Poiesis means to make the white blood cells, we call it what? We call it leukocytes, leukopoiesis. The final is thrombopoiesis. Thrombo means what? Platelets. Bala was telling you, thrombo means platelets. The formation of platelets is known as what? Thrombopoiesis. That is, poiesis means make. And remember, this is very important that thrombopoiesis happen through megakaryocytes. Megakaryocytes are large cells. Mega means large, karyo means nucleus, right? Megakaryocyte, large cell, and these cells fragment out. What happens? Fragmentation, fragments. And when megakaryocyte fragments, we get the platelets. That is, megakaryocytes breaks into fragments, we get the thrombocytes, which are the platelets. Now, next question, how can we separate? Simple question, how can we separate blood components? I told you blood has different components, but how do you separate them? How do you visualize them? Simple, centrifugation. Centrifugation, when you centrifuge, all the components get separated. Right, all the components get separated, centrifuge, and this is what happens. When you centrifuge a whole blood, keep it here and centrifuge it. What happens? All the different components will come into separate picture. And when you look at them, the major part of a blood is called as plasma. So blood is made up of red blood cells, white blood cells, and plasma. Now, when we look at this, and we also have the platelets now here. And this red blood cells, white blood cells, and platelets are called as what? Formed elements. They are called as what? Formed elements. These three are called as what? Formed elements. So white blood cells, platelets plus red blood cells are formed elements. Then you have the plasma. When we have the plasma, exactly, Bala is saying plasma is 55% in the blood. That is components of the blood. Let's understand the different components of the blood. Students, am I going too fast? Students, let me know in the chat. Just because, see, I have a mentality now. Now, I thought the time has been lost, so I might be going off a little fast. Students, let me know in the chat right now, am I going too fast for you? Or are you able to follow? See, some of the students will be watching this session for the first time, right? If you're watching it for revision's sake, it's understandable. But if you're watching it, if you're trying to understand the concept for the first time, is this session too fast for you? Students, let me know in the chat. I'm looking at the chat right now. Is the session too fast? Bala is saying, okay, sir. Is it correct? Is the pacing correct? Because I tend to go faster and faster. You have noticed by now, I tend to go a little faster when I'm excited. Is the pacing fine? Students, let me know in the chat. Let's understand the components of the blood. I told you, components of blood has plasma and formed elements. Now, what were the formed elements? WBC, RBC, and platelets. WBC, red blood cells, white blood cells, and what? The platelets are your formed elements. And then we have the plasma here. Plasma is technically the matrix. Can you see the matrix? Matrix nothing but is your plasma. So let's look into each and every one of them now. Let's understand what is the function, what are the different components of plasma. Ready? Amazing. So let's understand the plasma first. Let's understand the plasma. So plasma is what? Plasma is the straw color liquid. It is a straw color liquid which makes up the 55% of your blood. It is the 55% of your blood. 55%. 55% of the blood volume is plasma, which is a viscous fluid. See here. And what is plasma made up of? So I told you plasma is nothing but your 55%. Plasma is 55% of the blood. Now in this 55% we have divisions. Within the 55% we have multiple divisions here. Now let's look into divisions now. If you divide the plasma, if you take out the components of the plasma, we get what? We get the proteins, which are 6 to 8%. Less than 1% as the dissolved components, and more than 90 to 92%. From 90 to 92% of the plasma is water. So 90 to 92% of plasma is nothing but just water, but the main component here which you need to focus on here is the proteins. That is just 6 to 8%. So 6 to 8% of the plasma is proteins, and this is where the questions come from. So this is where the questions come from. See, that is, proteins are of three types here. Each and every protein is very important. Please listen very carefully now. The proteins, we have three types. We have albumins, globulins and fibrinogens. Albumins, globulins and fibrinogen. Now these three proteins are very important because questions come from here in the NEET examination. They'll ask you the function of them, they'll ask you the size of them, they'll ask you the composition of them. Okay, let's look into each and every one of them. Now before we understand the proteins, let's understand the dissolved components here. Here if you look at the dissolved components, we have different ions. We have sodium, we have calcium. Calcium plays an important role in clotting, right? It also plays a very important role where, students, let me know in the chat, what is the important role of calcium? What is the important role of calcium? Remember when we were learning about chloride shift in the last class, I think Kanika ma'am taught you that in the chloride shift, right? In the chloride shift we have what? In chloride shift, transportation of carbon. In transportation of carbon, calcium is very important, right? Now what about sodium? Where is sodium? That is depolarization. Sodium is used in depolarization. Mg2 plus is a major what? Cofactor in many reactions. Then we have your chloride. So this is involved in your CO2 transfer. Sorry, chloride is involved in CO2 transfer, not calcium. So not sodium. CO2 transfer, that is chloride shift. Then what H3PO3 also is involved in your osmoregulation. So all of this, fibrinogen, blood clotting, we'll get to that. We'll get to each and every one of them, but let me understand all of these. What are the dissolved components here? So we have different components. We have simple sugars, amino acids, lipids, urea, amino, carbon dioxide, oxygen, hormones and vitamins are involved in the dissolved component parts. But what is the most important part here is the plasma proteins. The most important part which you need to focus, all of you need to focus here is the plasma proteins. What are the different type of proteins? Let's understand the albumin first. Let's understand the albumin part. What is albumin? What is albumin protein? What is the role of albumin protein? I want each and every. Clotting and coagulation. Yes, yes. Coagulation means forming a mesh. Forming a mesh. And clotting, see, clotting is very specific to blood. Coagulation is the entire process. Coagulation of milk in a curd formation. Clotting is also a type of coagulation. Now here, albumin, I want all of you to write this down. Each and every one of you, so when you're writing, see, I might give you notes, but I've told you, when you make your own notes, that's the most efficient one. Please write this down. That is, albumin is the most abundant plasma protein. These are exact lines of your NCERT. Albumin is the most abundant plasma protein. What is the function? Function is the most important one. That is, it maintains the osmotic balance of the blood. Osmoregulation, exactly. Albumin, osmotic regulation. Clear? I hope this is clear. Osmotic regulation. Now what about globulins? Now what is osmoregulation? What is osmoregulation? What is osmoregulation? How does it do albumin? How does it do osmoregulation? Look here. When there is normal, in the normal cell, right, whatever the water molecules are there, right, water molecules are held by albumin protein. Albumin protein will hold the osmoregulation, hold the water molecules. But when albumin level goes down, if albumin level goes down, can you see water molecules are escaping outside. When there is not enough of albumin, water molecules will escape. And this is how osmoregulation has been done. This is how osmoregulation has been done. The next one is your globulin. Globulin, very important. Function is the most important here. Globular in structure, that is round in structure, includes immunoglobulins. Remember, IgG. IG, what is IG? In your human health and disease, we learned about acquired immunity. When you learn about immunity, we learn about IG, immunoglobulin. So IgG, IgM, right, IgG, right, all of these are immunoglobulins, includes immunoglobulins and carrier proteins. Primary function is defense mechanism of the body. So all of you should remember plasma protein known as globulin, which helps in what? Which helps in defense mechanism. Albumin does what? Osmoregulation. Globulin, defense mechanism of the body. For example, we have immunoglobulins. I told you, immunoglobulins, also called as antibodies. It shields our body from the pathogens. defense mechanism of the body. Now, last one is your fibrinogens. Fibrinogens, one of the clotting factors. It is fibrinogen is one of the clotting factor. Fibrinogen becomes fibrin, which helps in clotting. That is required for clotting of the blood. Fibrinogen becomes insoluble fibrin network. So fibrinogen, when it gets activated, when we learn about the clotting factor, when we learn about clotting mechanism, I'll tell you there. Okay, this fibrinogen will become what? Insoluble fibrin network, and this fibrin network forms a mesh, and this mesh is the result of your clotting. So clotting is done by what? Your fibrinogen, where fibrinogen gets converted to fibrin. Fibrinogen gets converted to fibrin, which helps in blood clotting. Amazing. I hope all of you know the functions of all the different three types of exactly largest protein. I hope all of you know the different functions. Functions are the most important here. Okay. Now, what is serum? What is serum? What is serum? So plasma, we just learned what is plasma. Plasma minus clotting factor. Very important. Write it down. Mark in your NCERT. Plasma minus clotting factor is serum. Easy. Plasma minus clotting factor is serum. I hope all of you should know that. Now we spoke about blood. What is blood? What is the formation of blood? So one more thing I forgot to ask you is, okay, we'll get to that. We'll get to the RBC there later on. We finished blood, we finished plasma. Plasma done. All of you know the plasma proteins? All of you know what is plasma proteins? Okay, let's understand the formed elements. I told you in formed elements we have what? That is, formed elements make up the 45%. Larger portion is what? Plasma, which is 55%, but your formed elements are just 45%, which includes WBC, RBC, and also platelets. That is, formed elements are what? Erythrocytes, which are your red blood cells, red blood corpuscles, leukocytes, white blood cells, and then thrombocytes, which are your platelets. Amazing. Let's go. Now, let's understand the red blood cells now. Are you following the pace, students? If you want me to decrease the pace, decrease the speed which I'm telling you, let me know in the chat, because I am very excited to this chapter. This is one of the most interesting chapter in human physiology, along with neural control. Okay, so let me know in the chat if you want me to reduce the pace. If not, we'll just go ahead now. So erythrocytes, I told you erythrocytes are your red blood cells. Formation of red blood cells is called as what? Formation of red blood cells is called as erythropoiesis. Poiesis means what? Formation, right? So also called as red blood corpuscles. Now students, who can tell me in the chat? Who can tell me in the chat why do we call them corpuscles? Who can tell me in the chat? Who can tell me in the chat why do we call them corpuscles? What is the meaning of corpuscles? Slow in heart part. Yeah, heart I'll go slow, don't worry. Heart I'll go slow, heart I'll explain properly. I'll explain you the cardiac rhythm today like never before. Trust me, cardiac rhythm, still students get confused. There are just 3 stages. Still people get confused, students get confused, but today we'll do it very clearly. Now, why do we call them corpuscles? What is the meaning of corpus? Meaning of corpus is dead body. Meaning of corpus is corpse. Corpse means dead body in English. Corpuscles because the nucleus has been removed. Right? The nucleus has been removed. Right? Remember, they are most abundant cells in the body, and this is the most important part. All of you should highlight in your NCERT. This is given in your NCERT also. The abundance, that is 5 million to 5.5 million. This number is also very important. This number is also very important, that is 5 million to 5.5 million. So each and every for your platelets, for WBC, very important. Think of RBC as the Ambani. Think of RBC as Ambani. Ambani is present. Ambani has a lot of money. So your RBC is present a lot. So in your blood, this is your Ambani. Right? This is your Mukesh Ambani. Okay. So this has a lot of 5 million to 5.5 million. Now let's understand RBC has a lifespan. RBC has a lifespan of 120 days. So RBC has a lifespan of 120 days. Now after completion of 120 days, RBC will die. And what is the graveyard of RBC? The graveyard of RBC is called as Spleen is the graveyard of RBC. Yes, in women it is 4 to 4.5. It is lesser in women because of the menstrual cycle, right? Now one more thing which you need to know here is, right, spleen. Now what exactly happens to the RBC when it dies in spleen? Who can tell me in the chat what exactly happens to the spleen in spleen? In spleen we have something called as, I'll write it here. So whenever the RBC dies in spleen, it forms something called as ferritin, and ferritin goes to liver where it is released as a bile salt or a bile pigment. Pigment. This is what happens to your spleen. So every single time the RBC dies here in the spleen, it gets converted to ferritin, which goes to the liver and it gets accumulated as a bile pigment. Exactly. RBC destroyed in the spleen. Now, that was your RBC. One more thing which you need to know about RBC here is very important. Very important. See, corpuscles means no organelles. What is the meaning of corpuscles? No organelles. What is the shape of your RBC? The shape of your RBC is biconcave shape. Here, what is the meaning of biconcave? Cave means what? A hole. So, if this is, one second. See here, think of this as your RBC. So RBC is cave. There's a small hole in the middle. Think of it as two of them and a small hole in the middle. That is your biconcave structure. See, a hole in the middle. And what is the thickness? That is 2 or 2.5 micrometer. Diameter length is your 7 micrometer. Disc shaped structure. Now one thing which your intro here is mammalian RBC are anucleated. That is no nucleus, has hemoglobin. So before that, one more thing which is new to need to know here is vada shape. Exactly, vada shape, similar to your vada shape. Now who can tell me in the chat, who can tell me in the chat the shape of RBC changes in some mammals such as your camels and llama, right? Llamas. In llamas and camels, the shape of RBC is little different there. The shape of RBC there is what? It is oval in shape. The shape of RBC is oval in shape in the case of where? The shape is oval in the case of camels. Very important to note here. Very important. Now one more thing I have to ask you here is, when we talk about hemoglobin, biomolecule students, hemoglobin is which type of protein? Hemoglobin is an example of which type of protein? Is it primary, secondary, tertiary or quaternary structure? Convex, amphibians, convex, yes. Now who can tell me in the chat? Biomolecules. Hemoglobin is a which type of protein? Is it primary, secondary, tertiary protein? Who can tell me in the chat? I'm looking in the chat. It is a quaternary structure. Which type of structure is hemoglobin? And hemoglobin has how many chains? Hemoglobin has how many chains? Who can tell me in the chat? Exactly, Vishal. Hemoglobin is a quaternary structure and it has 4 chains. 2 alpha chains and 2 beta chains. Amazing. This is your hemoglobin structure. Hemoglobin, 2 alpha chains and 2 beta chains. That hemoglobin, alpha containing protein. So remember, hemoglobin is a protein. It is a quaternary structure of protein which has 2 alpha chains and 2 beta chains. It carries respiratory gases, that is 2 to 16 grams of hemoglobin per 100 ml of blood. This is the exact line from your NCERT which I have taken. That is 2 to 16 grams of hemoglobin per 100 ml of blood. And this erythrocyte, which is your red blood cells, it has hemoglobin, that is 200 million molecules of hemoglobin per cell. That is your erythrocytes done and dusted. Now let's look at leukocytes. What are leukocytes? Leukocytes are your WBC. Leukocytes are your WBC. Let's understand the leukocytes now. Now, erythrocytes done. Leukocytes are your WBC. First thing which you need to know about leukocytes is they are colorless and white blood cells, and they lack hemoglobin. The hemoglobin is completely absent, and WBC has nucleus. That is, it is not written here, but WBC has nucleus, and the shape of the nucleus can vary. Shape of the nucleus can vary, and we'll get to that now. That is, lacks hemoglobin, less abundant. It is less abundant. This is like us. RBC was your Ambani. RBC was Ambani. This is like us, very poor. That is 6,000 to 8,000 per cell. Very less microliter of a blood. Just 6,000 to 8,000 cells, like us, very poor. No money. So it is 6,000 to 8,000 cells. But your RBC was how much? RBC was, red blood cells was 5 million to 5.5 million. Amazing. Now, short-lived. Remember, red blood cells has what? 120 days. This is short-lived. And it is protects from infection microorganisms. So, infectious microorganisms. The main production of WBC, the main work of WBC is for protection. Anil Ambani. Exactly. I am not going to say anything on that. Okay. So white blood cells, let's learn about white blood cells. I told you white blood cells has what? Nucleated. Can you see this is the nucleus here? That is nucleated, roughly spherical. It is roughly spherical in nature. It is nucleated and it is of two types. Now students, I'll just wait for 2 minutes. I'll just phagocytes. I'll tell you what is phagocytes. We'll come to that. All of you are so advanced. Trust me, I feel you guys already know the chapter. You're just here for revision. I know. So we have two types, granulocytes and agranulocytes. The meaning of granulocytes is with cytoplasmic granules. It is not without, it is small actually. Even in the case of agranulocytes, they have a smaller granules. They have granules, but they are very small. So this granules, because of this granules, we can stain them. We can do staining. Clear? We can do what? Staining. Granulocytes, agranulocytes with cytoplasmic granules, without cytoplasmic granules. Let's understand granulocytes now. Let's focus on granulocytes. That is the type of white blood cells. Let's understand each and every one of them. We have three main types. I'll tell you a trick to remember this. See, this is your B, eosin, neutrophils, benton. This was the cartoon which I grew up watching. From my 5th standard, I've been watching this cartoon. Benton is a series of about I'm not going to tell you about the omnitrix and all, but benton, that is basophils, eosinophils and neutrophils. Basophils, eosinophils and neutrophils. Benton. Remember, these are granulocytes. They are what? Granulocytes. And the thing about granulocytes is we can stain them. We can put color on them. We can play Holi with them. So all these can be what? They play Holi. So remember, Benton plays Holi. So all of these different granulocytes can be what? All these granulocytes can be what? Stained. Right? All these granulocytes can be stained. So one of the thing about eosinophiles is, remember, this percentage is very important. I want all of you to focus on the percentage here. That is, eosinophils is about 2 to 3% of total WBC, or of total WBC, 2.3 is your eosinophils. That is, eosinophils are acid loving. What is the meaning? I told you we can stain them. We can put color on them. And eosinophils take up what? They take up acidic dye. That is eosin. Eosin is a dye. That's the reason we call them eosinophils, because we can color them with eosin, which is a acidic dye. That is appears red in color. Here it appears red in color. And what is the function here? Function of eosin is very simple. Allergies. It is stopping the allergies. That is, have a role in stopping the allergic reactions, and also parasitic reactions. Also parasitic as well as this is a Dora the Explorer. Exactly. A parasitic reaction and allergic reaction. Every single time you have allergy, have role in stopping the allergic reaction. So eosin has what? Stopping the allergic and parasitic reactions. Now let's understand basophils. Basophils is just 0.1 to 1%. That's all. Basophils is just 0.5 to 1%. Eosinophils is 2.2 to 3% of total WBC. Now what? Let's learn about your basophils. They are base loving, meaning what? When you stain them, right? When you stain them. Sir, motion sickness or vomiting car, is it allergic? It's actually related to your brain. It's actually related, Vishal, it's related to your brain actually. The brain is not able to function properly in that moving. So it is not allergic basically. It is actually a brain thing. Allergy is mostly caused by an external stimuli. I think it can be, but I'm not sure. But car sickness is not allergic mostly. Now, basophils is base loving, that is they take up the basic dye. Example is your methylene blue. They can take up the methylene blue, which is the basic dye, and they look blue cool. They look blue, which is very cool. Next is your function of basophils. The function of basophil is they secrete histamines, heparin and serotonin. Histamines, heparin and serotonins are released by your basophils. Histamines mediate inflammation also, that is heparin natural anticoagulant. Heparin, which we know, heparin is a anticoagulant in our body. So heparin is a natural anticoagulant which makes sure that no coagulation takes place. What is your histamine? Histamine helps in inflammation, which helps in against inflammation. That's amazing. That's amazing. Kanika ma'am also told the same, because we have read it somewhere. Next person is neutrophils, which is the largest. Neutrophils is present in the excess. That is, neutrophils is 60 to 65%. 60 to 65% is neutrophils. That is the largest. And these neutrophils are phagocytosis. They help in phagocytosis. They take up and eat everything. That is phagocytic in nature. That is neutral loving. They take neutral dye, appear purple in color. What do? Neutrophils are phagocytic cells. That is, they engulf foreign material. Any foreign material, they eat it up. Phagocytes. That is, oh no, see, germ. The neutrophils will completely eat everything up. They are phagocytic in nature. See here, this is how your neutrophils will go. It will completely engulf. Here, a foreign body if foreign body is seen, neutrophils will completely engulf it. So let's quickly understand the revision of your granulocytes. Quick revision of granulocytes. Let's all of you please take a screenshot of this. That is, neutrophils take up the red and blue stains equally. Basophils take up the blue stain. Eosin takes up the acidic stain. Appear purple, blue and red. Function is phagocytosis. Function is mediates inflammation and also anti-inflammatory and also anticoagulant. That is your heparin. Then function is stops allergies and also parasitic activity by eosin. Those are the different types of granulocytes. We have just finished the granulocytes. Then we have agranulocytes. We have lymphocytes and monocytes. Now if you look closely here, neutrophils, I told you, neutrophils is 60 to 65%. Monocytes is about 6 to 8%. Eosin is 2 to 3%. Basophils is 0.5 to 1%. And lymphocytes is 20 to 45%. Now I want everyone to write this down. Or take a screenshot. Or write this down. Take a screenshot or write this down. Here. Done? Took a screenshot? Amazing. Now let's understand lymphocytes. Lymphocytes, very simple. Lymphocytes are of two types, T and B. Your B lymphocytes and T lymphocytes. Now who can tell me why are they called as B lymphocytes? Students, I'm going to see, I think there's a lag in chat. There is a solid 10 to 15 seconds lag in the chat because of some technical issues today. I'm going to wait for 10 seconds just like this, like a mannequin. Why are they called B lymphocytes? Why are they called T lymphocytes? If you have learned from Kanika ma'am, you should be able to answer this simple question. Why are they called as B lymphocytes? Why are these called as T lymphocytes? Let me know in the chat. Let me know in the chat. Why are they called as B lymphocytes? Why are they called as T lymphocytes? While you answer that, I'm going to sip some water. Why are they called as B and T? B lymphocyte secretes antibodies. Thymus. Bala is there. Bala is able to understand. See here. T lymphocytes because matures inside thymus gland. That is T for thymus, T for T lymphocytes. It recognizes the infection. T lymphocytes will recognize the infection and it matures in thymus gland. What about your B lymphocytes? It matures in the bone marrow. That's why the B lymphocytes, and the function of B lymphocytes is what? It releases antibodies. B lymphocytes recognizes the infection. T and your B lymphocytes releases the antibodies. Clear? It's clear. Are you able to understand now? Why it's called as T and B? That is region of maturation. So your B lymphocytes mature in the bone marrow. B for bone, B for B lymphocytes, T for thymus, hence T lymphocytes. Clear? Clear, students? Amazing. You learned something new. So that was your lymphocytes. Now let's understand the monocytes. Monocytes are large. They're very large in size. Monocytes are very large in size, and look at the nucleus. What is the shape here? Doesn't this look like headphones or your kidney shape? So this is kidney shape. Headphones is different. This is your kidney shape or horseshoe shape. Horseshoe shape or kidney shape. Monocytes. Monocytes transform into macrophages. The function of killing is mainly done by two different ones. That is, killing is mainly done by your neutrophils and here monocytes. Monocytes transform into macrophages like monster. Remember, M for monocytes, M for monster. M for monocytes, M for monster. So monocytes transform into your macrophages, which is a monster in nature. Here, this is how your macrophage will look like. Macrophage will keep on moving like your venom. It goes on, captures everything and eats it up. It's a monster. Macrophages are phagocytic cells. That is, they phagocyte, they kill the cells. Killing of cells is done by your macrophages. So complete revision here. Granulocytes. For WBC, granulocytes, eosinophils, basophils, neutrophils. Neutrophils is the larger in number, right? And neutrophils are phagocytic in nature. Monocytes are phagocytic in nature. Each and every function, I want all of you to write down in your book. Each and every function is very important. Each and every function here of each and every WBC can be asked as a question. That is T lymphocytes, B lymphocytes. Now this is a diagram from your NCERT, very familiar to you. Here, you can see the shape, monocytes larger in number. Here, horseshoe shape or kidney shape. Then if you look at basophils, they are small multinucleated. Here, if you look at basophils, they are nucleated. Can you say the eosinophil here? Color it down. Then if you look at T lymphocytes, then we have platelets and WBC. Clear, students? Is this clear to you? Function, each and every function should be known. Each and every function should be known, then only I can go ahead. Clear? Let there be carnage, exactly, let there be carnage. Next we'll start with thrombocytes. Is erythrocytes, leukocytes clear to everyone? Any doubt? Each and every function should be known. The concentration should be known. Percentage should be known. Percentage and function. Done? Can I go ahead? Next one is your thrombocytes, which is nothing but your what? Platelets. Thrombocytes. Thrombo means clotting. Cyte means cells. They're also called as platelets, and look at them. That is 15 lakhs to your 35 lakhs of platelets per microliter of your blood. So platelets are the highest in number. So if I ask you which is the highest in number, First is your RBC, first is your IBC, second is your thrombocytes, and the least is your WBC. Least is your WBC is the least one, highest is your millions. Ambani. Here this is us. This is your more rich, medium rich people, upper middle class people. That is formation of elements, thrombocytes. Let's understand the formation of thrombocytes. That is function is about blood clotting, releasing the thromboplastin. So your platelets release thromboplastin, and this thromboplastin helps in blood clotting. We'll understand the blood clotting later on, but for now just remember blood clotting is platelets release thromboplastin, which initiates the clotting. So that is the first part of the chapter is done. First part of the chapter is done. All the formed elements, plasma, everything done. So I want each and every one of you. I am not going to comment anything, Bala. Not going to comment anything. So I want each and every one of you to like the video. Before I go ahead, any doubt, if there is no doubt, everyone like the video right now, and I'll go to the next topic, that is your blood groups. We'll go to the blood groups. Everyone, students, let me know in the chat. Let me know in the chat the first part of the chapter is clear to you. Is the first part clear? Done? Then students, can I go ahead with blood grouping? So the previous part, all of you should learn the different names, composition and function. The questions mainly come from the function and the composition. Very clear? Amazing. Amazing. So everyone, if it is very clear, thumbs up in the chat and also thumbs up in the video. Let's understand the blood grouping. Let's understand the blood grouping. So blood grouping is what? How do we do the blood grouping? Very simple. The surface of RBC, that is your red blood cells. The surface of the red blood cells has what? Has genetically determined assortment of antigens. Very confusing line. Simply I'll tell you. These antigens are heredity, that you inherit the antigens. That is the meaning. And this RBC has these antigens, and we call them what? Agglutinogens. Agglutinogens. They are composed of glycoproteins and glycolipids. Agglutinogens is made up of glycoproteins and glycolipids, and where are they found? They are found on the surface of RBC. So your red blood cells, surface of red blood cells will have antigens, and always remember these antigens are heredity. You inherit them from your parents. Antigens are hereditary. See, antigens are substances that are triggered an immune response. We know what is antigens. Antigens are generally proteins, although some of their organic molecules can also be functions as antigens. So your RBC, red blood cells, on top of your red blood cells will have what? Antigens. Antigens will be present on your red blood cell. Based on the presence or absence of various antigens, blood categorized into different blood groups. So based on which type of antigen do we have, if you have antigen A, antigen B, if both antigens are present, or if no antigen is present, that's how we do blood grouping. That is, with a given blood group, there may be 2 or more different blood types. Okay, where is the blood grouping graph? Where is the diagram? I think, did I miss the diagram? Okay, before that, okay, it's there, it's coming. Okay. So we have, that is your first type, that is your blood grouping based on your antigen. The second type is your RH. ABO system and RH factor systems are the most important clinically. So ABO system is there, and there is your RH system. So let's learn ABO system of blood grouping. ABO system of blood grouping. So ABO system of blood grouping was given by whom? Your Karl Landster. So Karl Landster was in 1901 was awarded Nobel Prize on Physiology in 1930. Let's get skip to the main part here. So ABO blood group systems is based on 2 glycoproteins of antigens. So there are mainly 2 types of antigens, that is your antigen A and antigen B. If antigen A is present, blood group is A. If antigen B is present, its blood group is B. If both are present, it's AB. If nothing is present, it is O group. That's all. Genes coding for this antigens, they're not important for you. Here, people with antigen A. Students, all of you, people with antigen A. Yes, 29 years later. 29 years later, because recognition comes late. Recognition will always come late. People with antigen A has type A blood. Students, how many of you have type A in the chat? How many of my students are type A? Let me know in the chat right now. How many of you are type A blood group? How many of you are type A? How many of you are type A? Let me know in the chat. People with type A antigen, if surface there is type A antigen, your blood group will be A. If people with blood group B will have what? Type B blood group. That is surface has what? Antigen B. If both the antigens are present, it is type AB. Antigen A is also present, antigen B is also present. We call them type AB blood. Type AB blood have, your plasma has. Okay. What about type O? If both are not present, it is type O. Type O. So that is one concept. So guess my blood group. Who can tell me? Who can guess my blood group? Let me know in the chat if anyone can guess my blood group. It's very. Let me give you a hint. My blood group is a universal donor. My blood group is a universal donor. Guess my blood group. My blood group is universal donor. Okay. Bala is asking Bombay blood group. So there are different type of blood groups called as Bombay blood group and golden blood. There is golden blood, Bombay blood grouping. This is a Bombay blood grouping, basically has a different type of antigen, and this was first observed in your Bombay. It's O positive or not O negative. I have a RH positive. I have a RH positive. Don't worry, Bombay blood group is not in your syllabus. If you want to know what is Bombay blood grouping, I will make a short on it. Okay, so I told you there is a antigen. When there is antigen, we should have antibody. So antibody will be present in the plasma. So blood plasma contains antibodies called as agglutins that react with antigen A or antigen B if two are mixed. Now chat, who can tell me? Is this one, the antibody which is present in your blood plasma, is it inherited? Let me know in the chat. Do we inherit? I can give you blood bala. Is antibodies in the plasma inherited from the parents? Let me know in the chat. Is plasma, the antibody present in your blood plasma, is it inherited? I told you antigen is inherited, but is antibody inherited? Let me know in the chat. So there is anti-A. So always remember here, if your antigen A is present, right? If your body has antigen A on your body, if your antigen A is present on your body, if antigen A is, your blood group is A, means you have antigen A, then you will have antibody B. So always remember, if your blood type is B, you will have B antigen. Blood type is B, B antigen, but blood plasma will have what? Anti-A antibody. Why the opposite? The reason for it opposite is because if we have antigen B and antibody A, both will form and it will be killed. So that is the reason. If your blood type is B, B antigen will be present on your red blood cells. But what about the antibody? You will have anti-A antibody. You will not have anti-B because it should be opposite in your blood plasma. Now this is where your confusion gets clear. This is where all the confusion will get clear. Listen very carefully. If your blood group is A, if your blood group is said to be A, what is the antigen? Antigen will be A. What is the antibody? Antibody has to be opposite of it. That is antibody B. Now what is a donor? Means what? You can donate your blood to. You can donate your blood to. If your body has what? Antibody B, can you donate blood to your B? Can you donate blood to B? You cannot donate blood to B group. Why? Because if your blood has antibody B, this antibody B will react with your, what? It will react with antigen B. This will react with this, that is antigen, and will kill the patient. So when you're donating the blood, always make sure it should be the opposite. So when you're donating blood, if your blood has antigen B, you can only donate the blood to your A and O. You cannot donate the blood to B, because B will have what? B will have antigen B. If antigen B and antibody B react up, kill. Clear? Similarly here, if your body, if you have AB, if you have AB blood grouping, that you have antigens is both and you have no plasma, right? Can you receive blood from all of these? Yes, because if there is no antibodies present in you, if there is no antibodies present at all, I do not have any antibodies. So per people with AB, if a person has AB, right? If a person has AB, they do not have any antibody at all. If they do not have any antibodies, they do not have any type of antibodies, they can receive from all of them. So they are universal receivers, universal recipients. Receive, they keep on receiving. That is antibodies, they do not have any antibodies, so they can keep on receiving because they do not have any problem of antigen. Now what about your O type, my type of people? That is, we do not have any antigen on RBC. We do not have any antigen at all, right? We do not have any antigen. So, but what about the antibody? We have antibody for both A and B. We have the antibodies for A and B, but that's the reason if you take the blood from your A and B, we won't be able to survive because we have the antibody for A and B. So we can only take from O and donate. Clear? Any doubt here? Any doubt in this table? Any doubt in this table? See, only thing you need to focus here is antibody. If which antibody you have, depending on which antibodies we have, we can take the opposite of it. We can take the opposite of it. So if I have both antibody, this is me, this is your sir. Your sir has both antibody, A and B. Right? I have A and B. Can I take the blood from A? No, I cannot. If I take the blood from A, what will happen? My antibody will kill it. Person with O blood group, universal donor, is actually O negative. Person with AB group, universal recipient. See here, this is what he was telling. O minus. Let's understand RH grouping now. Universal donors are helpful to all in real life. Yes, we are universal donors, we can donate blood, but we cannot receive from all of them. We can only receive from O positive. I can only receive from O positive. So if I need blood, I have to contact Bala. Okay. So universal donors are helpful in real life. See, I'll tell you one thing. When you donate blood, right, they don't keep the blood as such. They will divide it into platelets, into your serum, into your factors, elemental factors. So when they divide like that, you need to be very careful when you're taking the blood. Let's understand RH grouping now. So RH grouping, the RH, the name RH comes from your rhesus monkey. That is rhesus, RH, rhesus monkey by Carl Landstein. That is RH antigen was first discovered. It comes from your rhesus. RH comes from rhesus. That is RH factor. Now same red blood cell will have RH factor on top. RH factor on top. RH antigen is observed on the surface of RBC nearly 80 to 85% of your humans. That is, if RH factor is present, we call them plus. O positive. So I have RH positive, so I have RH factor. O negative, they do not have any RH factor. A positive, they have RH factor. A negative, they do not have RH factor. Simple. It was discovered by Carl Landstein and Alexander S. Wiener in 1937. Genes of RH factor responsible for the chromosome number 1. So if it is present, it is RH is present, it is RH negative because your RH factors are absent here. And this is your complete blood grouping. That is A is RH is present, it is A positive. A blood grouping minus A negative, complete blood grouping is here. Next concept is erythroblastosis fetialis. All these are very easy concepts. I think all of you should understand this very easily. The main focus you need to have is the heart. The main focus in this chapter is the heart. The main focus is heart, the cardiac cycle and everything. Okay. I'm doing really well. I'm actually hungry little. I'm a little hungry because I had my lunch around 1 o'clock. I had my lunch at 1:30. So I'm a little hungry. Other than that, I'm doing really well. Sameer, welcome to the class. Can I start this topic? I think all of you would know, but I'll explain this very briefly. I'll explain this topic. Erythroblastosis fetialis. Let's understand with a simple diagram. This is the mother. That is, this is a case seen when your mother is Rh negative. That is, no Rh antigen. Our mother is Rh negative. Cygnus is doing well now. So if the mother is Rh negative, mother does not have any antigen. If the baby is Rh positive, so baby is Rh positive, mother is Rh negative. Now during the normal pregnancy, no problem. During pregnancy, there is no problem at all. During pregnancy, but during the delivery time, when the delivery is happening, see here, during the delivery, baby's Rh positive blood. Baby's Rh positive blood transferred to mother's bloodstream. Normally there is nothing. Okay, see here. So this is your mother. Mother is what? Rh negative. Baby is what? The baby fetus which is growing is Rh positive. The father should be also what? Obviously the father should be what? Rh positive. Now this is the first child. During the first child, if the baby is Rh positive, during the childbirth, some of the blood can get transferred to the baby. That is, baby's Rh blood cells enter the mother's bloodstream. So mother's bloodstream has what now? Mother will have both Rh negative and Rh positive. And due to this, what happens? Due to this, mother will produce what? Mother's blood produces antibodies against Rh antigen. Mother's body will produce the Rh antigen now. That is, Rh antigen is present inside the mother now. Rh antigen. So these antibodies against Rh antigen remain in the mother in the bloodstream. It will remain. So mother has what now? Mother has the antibodies. Mother has what? Antibodies for Rh. Mother has the antibodies for Rh through placenta. Yes, through placenta. Now, when the second pregnancy happens, during the second pregnancy, that is, mother's antibodies attack the fetus having the Rh factor. Now again, if the baby is Rh positive, the antibodies which was created in the starting will go and attack the blood cells. That can be fatal for the fetus. That is, it can cause anemia and jaundice, and this condition is called as what? This condition is your erythroblastosis fetialis. It will kill the baby. And what is the solution? Solution is very simple. All of you would know by now. We make our own antibodies. This antibodies during the first pregnancy, administration of antibodies, that is injection. We give them an injection after the first delivery. That is, this antibody will go fixed with the antigen. That is, administration of more antibodies. They'll go bind. That is, example is your rogum. Rogum is a type of your antibody which we administer. Because of that, mother can now survive. Mother can now what? Survive. Clear, students? Clear? This is what NCERT says. NCERT says anti-RH. NCERT says anti-RH, that is rogum. Rogum. Clear? Clear? Very easy concept. See, trust me, when you learn with visualization, right? When you learn with visualization, everything becomes easy. If I sit and write everything, you might get notes, but the concept will not get clear. That is the reason I don't write a lot. See, writing even you can do. But when you visualize and when you learn something visualizing, right, it gets stuck in your head now. Now you can, when you close your eyes, if you think of erythroblastosis fatty LS, you will think of the diagram which I showed you. The mother, the fetus is positive, the mother is negative, blood is getting transferred in the first time, antibodies are getting produced, right, and this antibodies will get affect the baby in the second pregnancy, right. So next is interstitial fluid. The next concept which you need to understand is interstitial fluid. Ready? The next concept is interstitial fluid. It's easy, it's very easy, trust me. This chapter is very easy. Now I'll show you I'll do a diagram for heart, okay? I'll do a simple diagram for heart which will make you understand what exactly parts of heart, what exactly is happening in heart also. Amazing. Now, next, interstitial fluid. Now, who can tell me what is interstitial fluid? Simple question. What is the interstitial fluid? What do we call the interstitial fluid? While you say that answer, I'll drink some water. What is interstitial fluid? What is this interstitial fluid? Where is it present? What is the function of interstitial fluid? Is it related to your lymph? I'll give you a simple hint. It is related to your lymph. Interstitial fluid is related to lymph. What is interstitial fluid? Anyone knows? Does Bala know what is interstitial fluid? Okay, Bala is saying it is present between the cells. That is one way to say it. It is present between the cells. Yes. Then anyone else? Anyone else can guess what is interstitial fluid? Can anyone guess the body fluid between blood vessels and cells? Amazing. Rakshi Ramireddy is saying it is present between the cells. Almost correct. It is Vaishali is saying outside the blood. Vishal is saying outside the blood. Let's see what is lymph here. This is your diagram which you need to focus on. See, this is your blood capillaries. Blood capillaries will release certain secretions, will release certain liquids. So blood capillaries will release certain secretions which will have water, WBC, very important, WBC and dissolved substances. So capillaries, blood capillaries will release certain substances, that is interstitial. This is your tissues. And this fluid, interstitial fluid is a fluid which is released from your capillaries, which is a leaking fluid. We'll get to that, why lymph is called as a tissue fluid. We'll get to that. This is the reason, Vishal. Interstitial fluid is leaking out. This leaking out will have WBC, dissolved substances and water. Now this fluid gets accumulated in a lymph vessel. All this fluid can be accumulated in the lymph vessel, and that is your lymph. So lymph is nothing but your interstitial fluid. Inside the lymph vessel is your lymph. Look at the diagram. Capillaries are releasing certain fluid, that is interstitial fluid. Interstitial means what? Between the cells. And this fluid gets accumulated in the lymph vessel, and this liquid is accumulating here, and we call that lymph. Lymph is nothing but your interstitial fluid. Accumulation of interstitial fluid is your lymph. So that's your lymph. Lymph is a colorless liquid, contains ions, nutrients, hormones, proteins and WBC. It is present in the lymphatic system, lymph vessels. It is present in the lymphatic system, that is lymphocytes. Remember lymphocytes, lymph. That is lymphatic system. This is your lymphatic system. Throughout your body will have the connection. Similar to how your blood connections is there, right? Blood vessels are connected everywhere. Similarly, we have lymphatic systems. That is, throughout your body we have lymphatic system, that is network of lymph tissues. Every single place, think of lymph system as your drainage system. Think of this as your body's drainage system. Every single time there is removal of waste, leaking out, every leaking out will be captured by your lymph vessels and they will be transported throughout your body. That is drain lymph into major veins, and what happens to that? Lymph will drain it into a veins later on. Here, network of lymph vessels collect lymph from all over the body and drain the lymph into the major veins. That's the complete cycle. Your blood vessels are releasing some interstitial fluid that is getting accumulated in the blood, in your lymph capillaries. Lymph capillaries form a network and they collect the lymph from all over the body and drain the lymph into the major veins. That is subclavian veins. It is draining here. Everything is getting drained here. That is subclavian vein is one of the example. Understood? Why is it called? Students, diagram is important. Diagram is important. Always in biology, you cannot just mug up. You cannot mug up the definition of lymph and all. You need to understand what is lymph. Why is the need of lymph? Why do we need lymph? Need lymph? No, lymph. Because we need to drainage. Every time there is a leakage in the body, the interstitial fluid is leaking. That is getting into lymph node and lymph vessels, and lymph vessels will dump it to your veins. Clear? Everyone clear? And I told you, interstitial fluid is made up of what? Interstitial fluid is made up of 20 to 25% WBC. I told you. And that's why they're called as lymphocytes. Lymph has what? Lymph has WBC. Lymphocytes. Lymph cells are called as your lymphocytes. We learned lymphocytes before. We learned about lymphocytes before. Remember lymphocytes? I was telling you lymphocytes in WBC. So lymph is what? Colorless liquid contains. This is done, I guess. Now, coagulation of blood. Next concept is coagulation of blood. Watch. Next concept, coagulation of blood. Let's understand the coagulation of blood very carefully. I will tell you one. I'll do one flow chart here. Try to follow this flow chart and then we'll look at what I have. Okay. Let's understand the flow chart here. I'll write it here, this side. Okay. So you have injury or trauma. Injury or trauma. NCERT says something called as trauma here. NCERT says something called as trauma. So whenever injury or trauma happens, what happens? Activation of platelets happen. Activation of platelets. Activation of platelet happens. Why is it going everywhere? Activation of platelets. Right. Activation of platelets. And this platelets will activate what? Platelets will produce what? I told you, platelets produce what? Thromboplastin. They produce what? Thromboplastin. This is B. Thromboplastin. And this thromboplastin will activate thrombokinases. That is the enzyme here. Thrombokinase. Now this thymokinase will do what? This thymokinase will help in conversion of, prothrombin. Prothrombin in the presence of calcium, prothrombin will become what? Thrombin. And this thrombin along with calcium here. This prothrombin along with calcium will convert your fibrinogen. I told you starting, we have learned all this. Fibrogen to fibrin. Fibrin. That is your injury, activation of platelets. Platelets will activate thromboplastin. Thromboplastin will activate thrombokinase. Thrombokinase helps in what? Conversion of prothrombin to thrombin. Now thrombin along with calcium will convert fibrinogen to fibrin. This is your complete coagulation of blood. Nothing more than this will be asked. Simple. Now let's understand what is there. Let's understand what is the blood coagulation again. Injury is happening somewhere. Right, injury is happening, damaged tissue outside the vessel, cascade of clotting factors. Cascade means what? One after the other event is happening. One after the other event is happening. That is cascade. One after the event is activation happening. That is platelets in damaged vessels, cascade of clotting factors. Platelets will produce what? Platelets will produce thromboplastin. And thromboplastin will produce thrombokinase. Thrombokinase activates prothrombin activator. That is your thrombokinase. Thrombokinase is your prothrombin activase. That is your thrombokinase. If prothrombin gets activated, prothrombin gets converted to thrombin with the help of calcium, and when thrombin gets activated, it converts soluble fibrinogen to insoluble 3D form fibrin matrix, and this 3D fibrin matrix, this one here, it will go and sit on the form of mesh. This will form a mesh. Fibrin matrix will form a mesh, and that will result in the clot formation, thread fibrin. That will result in the clot formation. The major element which is important, ion which is important here is calcium, Ca2 plus. Ca2 plus. One more thing which you need to know here is, sir, internal bleeding. So bleeding which happens inside, obviously. Flowchart was there, right? Bala, flowchart was there. This is the flowchart. This is the flowchart which you need to understand. Here, first one, flowchart. This is the flowchart which you need to follow. Injury, activation, platelets. Platelets will activate thromboplastin, which will activate thrombokinase. This thrombokinase is the enzyme which will which help in conversion of prothrombin to thrombin. Along with calcium, fibrinogen gets converted to fibrin. That's all. This is your blood clotting, complete blood clotting. One more thing which you need to know is heparin. Now students, who can tell me heparin is secreted by which? Heparin, I told you, heparin was secreted by which one? Which? Where did we study heparin? Where did we study heparin, students? When I was telling you which topic, did we study heparin? Heparin. Basophils. Because external, if it happens, it will happen on the skin. Internal damage can happen because the blood vessels blood capillaries will burst. If blood capillaries burst, bleeding will happen inside. Blood capillaries are bursting, right? So bleeding is inside. Basophils, exactly. It's written here only. Oh my God. Erase this. So it's produced by mast cells and basophils, which will help in what? Anticoagulant. They will not sure coagulation happens. It is written here only. Now, one more thing which you need to know here is one more anticoagulant, which is nothing but your EDTA. How many of you heard it? EDTA. I do not know the full form. I do not remember the full form, but I remember teaching EDTA before. Now, EDTA, what will EDTA do? How does EDTA do? EDTA is an artificial. EDTA is an artificial anticoagulant. How do they function? Function is simple. This is used in your blood banks. This is used in the blood banks. Blood banks. How do they function? Simple. EDTA will capture the calcium. EDTA will capture the calcium. If calcium is being captured, will there be clotting? Obviously not. There won't be any clotting. There won't be no clotting. That is, clotting agent has been removed. So this is your EDTA. Clear? Students, clear? This concept is clear? I don't know why this is coming again. Is the concept clear? Is clotting clear? Is clotting clear? I have cold now, little. It's clotting clear, students? Because after circulatory system, we have to go understand what is circulatory system, and then we have to learn about arteries, veins, heart. What is it of how does the cardiac rhythm works? What is lub-dub? Right? We have to learn about what is different volumes. Someone actually go. It's called ethylene di. Amazing. Why is my mother calling? Can I start circulatory system? Can we start with circulatory system, students? Okay, take 5 minutes break. I will take the 5 minutes break now only if you want. Circulatory system will start in 5 minutes. Well, I'll start circulatory system in 5 minutes. Students, let me know in the chat. Can I start circulatory system in 5 minutes? Because this is going to take a long time. We have open circulatory system, closed circulatory system. Then after we have learned about. Okay, take 5 minutes. The time is now 9:05. 9:10 will start. 9:10 p.m. At exactly 9:10 will start. Okay, 9:10. Okay, 9:10. Yes, I remember. Why so many accounts? Krishnaveri. I remember you came from Harshita account also, right? I hope you're doing really well. Do you remember Unthinkable? Yes, it's me. I remember. Second treatment, I'm scared. I have no more to share. I'm sharing here, sir. No, it's fine. It's fine. I hope you're doing really well. I hope you're doing really well. Just wait 5 minutes. I'll just drink some water and come. I'll sit for 5 minutes and we can start. Five minutes will start. Where I'm from Bangalore. I'm from Bangalore. Oh, it's my mom's phone. Okay. You'll do well. Don't worry. Don't worry. You'll do really well. May the God be with you. May the Ganesha. I believe in Ganesha a lot. May the God be with you. I'm chilling only. This is my chill time. Don't worry. This is my chill time. I have to just call my mother. 2 minutes. I'll just talk to my mother and come. Mama. Yes, ma. Class number 3. Class number 3. Check, check, check. Can we start? Can we start, students? Amazing. When I got the break, all of you got the break. Can I start? Let's start. Amazing, all of you are here. The break is over. Okay, let's start. So if I can start, I want everyone thumbs up in the chat if I can start. Thumbs up in the chat, everyone, thumbs up in the chat if I can start the session. Thumbs up in the chat if I can start the session. I want to see a lot of thumbs up. I want to see a lot of thumbs up in the chat and I'll start the session. Amazing. I'll just keep my phone aside. Yes, amazing. So yeah, let's talk about circulatory system. Simple question. Let's start with simple question. Humans, chordates. Humans, do humans have open system or closed system? Simple question for all of you. Do human beings have open system? Do humans have open system or closed system? Who can tell me in the chat? Do humans have open system or closed system? Let's understand the open system first because closed is seen in humans. Open system. Now look at open system. The first thing which you need to notice here is in open system case there is no capillary bed. That is open circulatory system. The fluid is called as hemolymph. The fluid which is transferring here is called hemolymph, which is pumped by heart through vessels. From heart it is getting pumped through vessels and it is directly opening up. It is going into an open space, that is vessels open into open space or body cavity. Right, vessels are directly opening up and this is your open system, that is heart, blood vessels and body cavity. There is a complete absence of, in the case of open system, there is a complete absence of blood capillary bed. So when there is no capillary blood, the blood which is directly releasing into open space. That is the case which is seen in your open circulatory system. And here look at this. This is mostly case of your arthropods. And the opening is called as ostia. The opening is called as ostia, that is from heart whatever the blood is being pumped, right, it is directly releasing into the open system. It is directly into open system. But in the case of your closed system, the blood, see here, presence of blood capillaries, that is your body cells, it is not opening to body cells. In the case of your closed circulatory system, we have capillaries here. There is a capillary blood, that is blood pumped by the heart is always circulated through closed network of blood vessels. So in the case of closed circulatory, we have the blood vessels, and in the case of closed, we can regulate the blood. Double circulation, we'll come to double circulation later on. Now advantage here is flow of the fluid can be regulated. So in the case of humans, blood regulation can be regulated because it is closed. It is blood capillaries are being present here. Capillary blood is present here. Example is your annulates. Even in the case of annulates, the blood is getting closed. Basically, think of it like a circuit. If the circuit is closed, it is closed. If the circuit is open, it is open. Even in the case of humans, chordates, that is we have heart, blood vessels, right? Heart and blood vessels. That is we have human circulatory system is very advanced. In the case of human circulatory system, we have different components such as your circulating fluid, such as your blood. Then we have a heart. Then we have blood vessels. Throughout our body, we have the blood vessels. We have a human heart which is pumping the blood. Then we have the circulating fluid which is your blood. Okay, so let's talk about circulating fluid. which is done. We have already talked about circulating fluid, which is your blood and lymph, fluid connective tissue, transportation of oxygen. Lymph is colorless fluid, carries immune cells, that is lymphocytes and nutrients. Then we have the heart, and let's talk about the heart now. Let's talk about the human heart. Let's talk about the human heart. So human heart is said to be a I'll get to that. Double circulation, incomplete cerebral circulation. I'll get to that when I talk about your in case of your amphibians, in case of your birds, what happens. I'll get to that, not right now. After the heart, I'll teach you that. After the heart is done, I'll teach you that. Okay. I'll talk about what happens in birds, 2-chamber heart, 3-chamber heart. I'll explain that later on. Let's understand heart first. That is there. So heart is a muscular organ. Do we heart? We say heart is a myogenic because it has a rhythmic contraction. It happens, contraction happens on its own. So even if you remove this heart, out of your body and keep it on the table, it will still be pumping. It will still look like it's moving, right? Why? Because it has what? Why? Okay, there's one simple thing why which makes the heart, because we have the signals, that is SA node, everything is there. Because of that, the pumping is there. That is, we call it automatic creation. It is rhythmically, it is moving. It is automated. It is myogenic in nature. It keeps on moving. Contraction is there. Even if you remove it outside, that is, the size of the heart is almost like your clenched fist. This is the size of your heart. This is the size of your heart. Now, heart is made up of 3 types of layers. That is your, this is not your syllabus. That is pericardium, myocardium, endocardium. That is the layers of the heart. But what you need to focus here is the structure of the heart. I will draw the structure of heart from my side. Then let's understand the structure of NCERT heart. Clear? Let's understand the structure which I know. Then we'll look at the structure of which is given in your NCERT. Clear, students? Now let's understand the structure of the heart. I told you structure of the heart is made up of. The heart is made up of 4 different chambers. The heart is made up of 4 different chambers. The first 2 chambers are smaller in size, that is your right atrium and left atrium. Right atrium and left atrium are a little smaller in size, but your right ventricle and left ventricle, right ventricle and left ventricle are little larger in size. Now to this right atrium and left atrium, we have certain, what do I say, capillaries coming into the picture. For example, in case of your right atrium, it receives deoxygenated blood. Alright, here it receives what? Deoxygenated blood. Listen very carefully, please write this also in your syllabus. It receives deoxygenated blood. Right? From where? From where do this right atrium receives the deoxygenated blood? It receives deoxygenated blood from 2 different types, 2 different places. It receives the deoxygenated blood from your superior vena cava, and also it receives the blood from your inferior vena cava. From here, this is your inferior vena cava. What is the difference between superior and inferior? Above the diaphragm, if whenever the blood is coming from above the diaphragm, it is from superior vena cava. Below the diaphragm, from inferior vena cava. All the deoxygenated blood is going to your right atrium. Right, going to the right atrium. I want all of you to draw this. Now, blood from your right atrium is going to right ventricle from here. Let me change this. Blood is going like this from your right atrium to right ventricle. Now from right ventricle, blood is leaving. From right ventricle, the blood is leaving. The blood is leaving towards this structure right here. Now this structure right here, this part is called as a pulmonary trunk. This structure right here is called as pulmonary trunk. Here, this one. This right here is called as pulmonary trunk. It's called as what? Pulmonary trunk. And these 2 parts, this will go to your left lung, right, this will go to your left lung, this will go to your right lung. through this structure. This is your pulmonary artery. Pulmonary artery. Write down. I want all of you to draw this diagram. This is your pulmonary artery. So deoxygenated blood is coming from where? Superior vena cava or inferior vena cava? Inferior vena cava below the diaphragm, superior vena cava above the diaphragm, right? Above the diaphragm. Now deoxygenated blood is coming, right? From your, first it enters through right atrium, then it goes to your right ventricle. From right ventricle, deoxygenated blood is leaving to pulmonary artery, to different lungs. That is, from right ventricle it is going to different lungs. That is, left lung or right lung, through pulmonary artery it is exiting. Now what about your right atrium? Left atrium. Left atrium will receive the blood. Left atrium will receive the blood. It has 4 tubes here. Pair of 4 tubes. This 4 tubes right here is called as your 2 pair of pulmonary veins. 2 pair of pulmonary veins. Now these pulmonary veins will bring oxygenated or deoxygenated blood. Pulmonary veins will bring oxygenated blood from the lungs. So this is going to be what? This is going to be your oxygenated blood. Oxygenated blood. So oxygenated blood is coming from where? From lungs through pulmonary veins into left atrium. So left atrium has what? Oxygenated blood. Now from left atrium the blood is transferring to the left ventricle. From left ventricle the oxygenated blood is releasing to, from your left ventricle blood is leaving with the help of a tube called as here is your aorta. Now this aorta right here is the largest artery. Now one more thing which you need to know here is this is the basic structure. Deoxygenated blood is coming through superior vena cava, inferior vena cava to your right atrium. From right atrium deoxygenated blood goes to right ventricle. From right ventricle it is transferred to your pulmonary artery. To pulmonary artery it goes to the lungs. Now from one lungs it gets oxygenated. Now once it gets oxygenated it is entering through pulmonary veins into your left atrium. Now from left atrium it is going to your left ventricle. Now from left ventricle it is going to, now it is oxygenated blood. Who needs oxygenated blood? All the different body parts. All the different body parts need oxygenated blood. So this is aorta, through the aorta all the different body parts will receive the oxygenated blood. I will tell you again before I go to other concepts. Before I go to the other concepts as your valves and other things, let's understand this properly. First, the blood is entering where? The blood is entering to right atrium. Blood is entering through the right atrium. So which blood is coming? Deoxygenated blood is coming. Blood does not have any oxygen. It is entering through superior vena cava and inferior vena cava. Superior vena cava above the diaphragm, inferior vena cava below the diaphragm. Now it is entering here. Now from here it is entering to your right ventricle. To the right ventricle, deoxygenated blood. Now from deoxygenated blood it is entering to right ventricle. From here it is leaving to different lungs. It is deoxygenated. It needs to get oxygenated. So it is leaving through pulmonary artery to different lungs. Now from the lungs one it is oxygenated. It is entering to your left atrium through pulmonary veins. Through pulmonary veins which are 4 in number. There are 4 pulmonary veins here. Pair of 2. There are 4 pulmonary veins here. It is entering to your left atrium. From left atrium oxygenated blood is entering to your left ventricle. From left ventricle it is leaving outside. It is leaving to different parts of the body with the help of artery. Which artery? Aorta. Aorta is the largest artery. Aorta is the largest artery. Clear? I hope till here it is very clear, because now I'll talk about the valves. I'll talk about valves now. Till here it is clear, students? I hope till here it is clear. Now I'm going to talk about valves. Now valves are your muscular flaps. Valves are muscular flaps. These muscular flaps will make sure unidirectional flow of blood. This will make sure what? Unidirectional flow of blood. and we have different type of valves here. The first valve which you need to focus here is, the first valve is coming from your, where? Inferior vena cava. I'll change the color to green. The first valve is here. The first valve which is here from inferior vena cava, the name here known as what? It's, anyone know? It is called as what? Eustachian valve. Eustachian valve, which is present here in the side here. This valve is present in the side here for the inferior vena cava. For superior vena cava, there is no valve. For superior vena cava, there is no valve. So first valve is here. Now second valve is here, present between here, this transportation here. Now this valve is nothing but your tricuspid valve. Tricuspid valve. Write down this. If you do not write down on practice, this will never go to your head. If you do not write down on this, this will never go to your head. Tricuspid valve. Now what about this valve here? From left atrium to left ventricle. Now this valve is your bicuspid valve. Bicuspid. Bicuspid valve. Why bi and tri? Because there are 3 flaps here. It is made up of 3 flaps. When I show you the NCR diagram, I'll tell you. Bicuspid valve is it's also called as mitral valve. Exactly. Bicuspid on the left. Okay. Bicuspid valve. It is made up of 2 flaps. Now next one. Now this pulmonary, it is going out. Deoxygen blood is going out here, right? Here. One more valve has to be here. One more valve has to be here. And the shape of this valve is semi-lunar. And we call them semi-lunar valves. These two right here is semi-lunar valves. Clear? Now one more thing which you need to know here is the what? Muscles. One more thing which you need to note here is the muscles. Let me change the color to red. No, red is already here in use. Let's use this color. Now one more thing which you need to notice here is, yes, mitral valve, semi-lunar valves. Done. Now one more thing which you need to notice here is, this left ventricle is very thick. Left ventricle is very thick. Left ventricle is very thick in nature. Why do you call it so? Why do you saying it very thick? Because of there's muscles here. Which muscle? Papillary muscles. Here is something called as papillary muscles. Papillary muscles surrounding. Surrounding is your papillary muscles here. Now one more thing you need to know here is the holding of this papillary muscles to here. One joining is there. Now this joining, can you see one joining? You will see this in your NCR diagram also. You will see this part in your NCR diagram also. Now this, can you see this extra thing? This is your collagen fibers. These are what your collagen fibers. It is holding, collagen fibers are holding the valve, bicuspid and tricuspid valve. This in your NCR is what? In your NCR it says chordae tendinae. Chordae tendinae is the, see, can you see your fibers which is holding? White are fibers. This is your, this one, chordae tendinae. This is your collagen fiber, this one. Can you see? I'll do it, I'll make it white. I'll make this white. Here, this is holding the valves. This is holding the valves to make sure the valves remain close. To make sure chordae tendinae makes sure what? The valves remain close. Now, anything which I'm missing here? One more thing which I'm missing here is the what? I'm missing the layers between. Now this middle layer, I don't have space to write here. So I'll just write it here. Now this is your, I told you this is your 2 ventricles. Now between the 2 ventricles also thickness is there. Between the 2 ventricles thickness is there. Now this thickness here is called as what? This thickness is called as what? This thickness is called as thick muscular interventricle. Inter means between, ventricular, between two ventricles, interventricular septum. Interventricular septum. Now what about this septum here? Here also septum. Now this septum here is called as what? This septum here, between this and this is called as atrioventral septum. This here is called as atrioventral septum. Here, this one, middle, between the arteries, between the arteries and ventricles, between your atrials and ventricles, is called as what? Atrioventricular septum. This one. Atrioventricular septum. Is there a septum present here also? Yes, there is a septum present here also. This we call as intermuscular interatrial septum. This one here, but this one is between these two. This is called as atrioventricular septum, and this is made up of not muscle. This is made up of your fibrone, thick fibrous layer. This is made of fibers. This is fibrous in nature. This is fibrous in nature. Both of this and this is muscular in nature. Students, are you able to follow, or are you getting confused somewhere? Are you able to follow, or are you getting confused in the entire structure? Is anything missing here? This is the structure of your heart from my side. This is the structure of heart from my side. Anything here? This is the structure of heart from my side. This is how you can also learn, but this is how you can actually learn. Any doubt here? Blood is coming from where? All the valves, all the septums and all the linings, this white color part, choroid tendineae. Fine? Clear? Can I go ahead? So this is how you should also study. This is how also you should study. Very simple diagram. If you draw, if you watch the video again, if you replay it again, you should be able to draw the structure. Now let's see what NCERT says. Let's say what NCERT says. So NCERT, let's see this side. Let's see this side. Aorta. I told you aorta here. Aorta is what? Aorta will take what? From left ventricle. It is coming out from left ventricle, and it is what? It has oxygenated blood. So aorta has what? Oxygenated blood. Then it is coming from your left ventricle. It is coming from left ventricle. It is getting released from left ventricle. Now here you have bundle of that will get to later. The pulmonary artery. Where is pulmonary artery? Pulmonary artery is here. Where is pulmonary artery? Pulmonary artery is here. What does pulmonary artery carry? Pulmonary artery carries the deoxygenated blood from your right ventricle. Pulmonary arteries carries the deoxygenated blood right here. Deoxygenated blood. Oxygenated blood. What about pulmonary vein? Pulmonary vein, I told you, they're present in many, like this pairs. They're present in pairs. I told you they're present in pairs. Pulmonary veins. Pulmonary veins coming from top. Pulmonary veins will bring the oxygenated blood into your left atrium. That is pulmonary veins to the left atrium. Then we have interventricular septum, middle, interventricle, between the two ventricles, septum. This is the apex. Meaning what? The meaning of apex is what? The meaning of apex is what? Apex means your heart is pointing towards the left. The end of it is pointing towards the left. Then you have right ventricle. Right ventricle is sending what? From right ventricle, can you see? From right ventricle, your pulmonary artery is actually going out. Then you have atrioventricle node. Nodes I'll get later. Nodes later on. Right atrium. What happens in right atrium? In your right atrium, from your superior vena cava and inferior vena cava, deoxygenated blood is going into your right atrium. So toward right atrium, superior vena cava is releasing the deoxygenated blood. Yes, it is completely about heart. Clear? Now here one more thing which you need to notice is, I'll get to this later on. that is your SA node, and your AV node and SA node. I'll briefly explain AV node and SA node. We can understand in detail later on. I'll briefly explain you what is SA node and AV node. Now think of SA node as your king. Think of SA node as your supreme leader. Supreme leader will give out a command. Supreme leader will provide a command. And that command will be taken up by AV node. Supreme leader is your SA node. SA node is the supreme leader which is giving a command. Who takes the command? Command will be taken by your next captain. Supreme leader, captain is taking the command. Now from SA node, the information will be sent to your normal soldiers. From your SA node, AV node will give the information to your soldiers. Soldiers are your rest of the cells, rest of your blood cells. So that's why this SA node is called as what? Pace setter. It's the king. Pace setter. Pacemaker. Your SA node is pacemaker. Pacemaker. I'll get to that. Why it's called pacemaker. I'll get to that later. SA node is your pacemaker. It is a king. It will give the command. Command will be taken up by your AV node. Here, AV node will take up the command. It is your captain. From the captain, the rest of the soldiers will get the command. That is your bundle of Hess. From here, can you see? From here, it is coming. Bundle of Hess is coming here, and it is getting separated here. Separated here. And this separation is nothing but your Purkinje fibers. Is nothing but your Purkinje fibers. Clear? But SA node, AV node, bundle of Hess, and Purkinje fibers are coming outside somewhere. Now, can you see here? 3 flaps is not shown here. Never mind. Clear? Is this clear, students? Is this clear? Anything left here? Anything is left here? I told you this is your cord tendons is here. This, which helps in what? Which helps in holding back the valves, bicuspid and tricuspid valves. Structure of heart is clear. We will get to the structure of heart after this. Is structure of heart clear? Any doubt in structure of heart? Structure of heart should be clear to you. You try learning the concept wise, which I taught you. Try learning the concept one. The diagram one will be easy to you. For your diagram will be only important for your naming. But the box one which I showed you will be able to understand. The concept will get clear when you learn the box one. Okay. Amazing. Amazing. Clear, Ravindrani? Clear, Bala? Is that clear? Now, before you understand the cardiac movement, cardiac rhythm and everything, how the heart works, heartbeat, cardiac cycle and everything, let's understand the circulatory system. That is veins, capillaries and arteries. Quick look at veins, capillaries and arteries, then we'll understand how the heart works. Okay. Now, I told you human circulatory system, that is your arteries and veins has 3 layers. Your inner layer is called tunica intima, that is squamous epithelium. Then middle layer is your median, that is tunica media, smooth muscles and elastic muscles, and external is externa, tunica externa, fibrous connective tissue with collagen fibers. This is not that very important, but very important part here is the difference. That is your arteries and veins. Can you see the main difference between arteries and veins is the lumen? Can you see the lumen is small here, lumen is larger here in the case of veins? Now who can tell me why is the lumen larger here? Why is the lumen larger in the case of veins? Who can tell me in the chat why is the lumen larger in the veins here? And what does your arteries do? That is arteries from heart. Arteries will take the blood and different parts of the body. So arteries carry your oxygenated blood. They travel from your heart to different body parts of the body. Arteries will take. Then we have from different parts of the body back to the heart is taken up by your veins. That is arteries carry the blood away from the heart. Away from the heart, veins carry blood towards the heart. Pinkish or red in color, deep blue in color. Here, arteries and veins, thin wall blood vessels which help in exchange of material at cell and the body level tissue. Deoxygenated blood. Yes, 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 yes. Arteries carry oxygenated blood. Arteries carry oxygenated blood away from the heart, and veins carry deoxygenated blood from the towards the heart. So one more thing I want to ask you here is, veins receive deoxygenated blood. Veins carry deoxygenated blood from towards the heart. This is a very simple, you would have learned this in your lower classes. I don't think this is very important. All of you would have learned this in lower classes, that in class 8th and 9th. That is, arteries carry oxygenated blood, veins carry deoxygenated blood. Now one more thing I want to tell you, there is the pumping of blood. When I come to pumping of blood, I'll tell you the why aorta the largest. Okay, simple. All this is very simple. Now one more thing which you need to notice here is the complete circulatory system. That is your from right ventricle, what happens? Right atrium, right ventricle to pulmonary artery to the lungs. Now think of this diagram as what I drew. When I drew the diagram, I told you this, all this. Your right ventricle, pulmonary artery, it's going to pulmonary artery. Down I drew, remember, I drew it in the down. That is right ventricle, pulmonary artery, lungs it is going. Once it gets reformed in the lungs, it's going to pulmonary veins. From top, pulmonary veins were coming. Remember, pulmonary veins were coming from left atrium to left ventricle to aorta. Aorta is the artery. From aorta it goes to the tissues. From tissues it goes to vena cava. Again, vena cava will take all the deoxygenated blood and it will travel to the right atrium. See, this is the most basic thing which you need to understand. Very basic structure of blood flow. Now this part here, from lungs to the from lungs to the ventricles and atrium, lungs to the heart is called as pulmonary circulation. From the tissues to the heart is called as what? From the tissues to the heart is called as systemic circulation. Pulmonary circulation, systemic circulation. That's all. Clear? Any doubt there? I don't think anyone of you should have doubt there. It's the most basic thing which you need to follow. Now this is where things get interesting. Bala was asking, remember, types of circulations. Bala was asking us, what is single circulation? What is incomplete circulation? Right? Bala was asking this. What is single circulation? What is your incomplete double circulation? Right? Incomplete double circulation is here. What is incomplete double circulation? What is complete double circulation? Let's understand that before we understand the working of the heart. Yes, 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 yes. Which one, Ravindrani, which one? I'll explain this concept will explain now. Which one? Ravindrani, which one? Which concept? The movement of blood. This one. This one. This one. This one. Ravindrani, this one, you want to repeat this one? Which one? Which concept? Tell me which concept which needs repetition. I'll explain that. Which concepts need repetition? Circulation. Here. I told you your right atrium. Sorry, let's not atrium. Let's go to this one. I'll draw the diagram here and I'll explain again. Here, when I did the boxes, remember I did the boxes here. When I did the boxes, I drew your right atrium here. What was here? This is your right ventricle. What was here? Left atrium. What is here? Left ventricle. Now I'm telling you, from vena cava, blood is going to right atrium. From vena cava, from here, superior vena cava or inferior vena cava, it is going to right atrium. From right atrium, where is it going? From right atrium, it is going to right ventricle. From right atrium, it is going to right ventricle. Now from right ventricle, where does it go? From right ventricle, it is going to the, where? Pulmonary arteries. Here, pulmonary arteries. Pulmonary arteries to where does it go? From pulmonary arteries, it goes to where? It goes to lungs, I told you. It goes to the lungs. It goes to the lungs. It goes to the lungs. Now from lungs, from where it goes from lungs, using pulmonary vein from here top, I drew pulmonary vein. With the help of pulmonary vein, it is going to left atrium. Here, left atrium. From left atrium, it goes to left ventricle. Left ventricle. From left ventricle, where it goes to? Aorta. Here, aorta. Aorta. From aorta, it goes to tissues. It goes to tissues. So that is the entire flow which I drew here. Now, when pulmonary circulation means what? Whenever the blood is passing from your right ventricle to left ventricle and to lungs, whenever the blood is passing through the lungs, it is called as pulmonary circulation. When the blood is transferring from lungs, when lungs is involved, it is called as pulmonary circulation. And when your tissues system is involved, other system of the body is involved, such as your different cells, when different tissues are involved, we call this systemic circulation. Blood passing through the system is called as systemic circulation. Blood passing through the lungs is called as pulmonary circulation. It's called as what? Pulmonary circulation. Blood passing through the lungs, here, this part is called as pulmonary circulation. Clear? Clear? Ravindrani, clear? I hope this is clear, Ravindrani. If this is not clear, let me know in the chat right now. If this is not clear, I'll explain this again. Is this clear, Ravindrani? Bala, is this clear? Can I go to circulation? Can I talk about circulation? Types of circulation. Clear? Amazing, amazing. All of you are amazing. Now, types of circulation. We know we have birds. Birds have what? Never mind. Your birds have what? 2-chamber heart. In case of your fishes, birds have 4. Remember. So, in the case of your fishes, there is only 2-chamber heart. Only 2 chambers. In humans, we have 4 chamber. 2 atriums and 2 ventricles. But in the case of fishes, there is only 2 chambers. 1 atrium and 1 ventricle. So heart has only how many chambers? 2 chambers. So whenever the blood is transferring from gills, when the oxygen is transferring from gills, it goes through the heart of one. From it goes to the atrium, it comes out of the ventricle. Direct. There is always mixing of blood is happening here. Mixing of blood is happening here in the case of single circulation. In case of single circulation, there is mixing of blood. Complete mixing of blood. Complete mixing of blood. See here. So if this is body of the body, this is the body of your fish, and this is your gills, and this is your heart. Heart, gills, body. From your heart, from the heart, deoxygenated blood is going to your gills. Deoxygenated blood is going to gills. From gills to the body. From gills to the body, oxygenated blood is going. From gills to the body, oxygenated blood is going. Again, when the body gets consumed, it is consuming all the oxygen to the heart. To the heart, all again deoxygenated blood is going. Simple cycle. Your circulatory system in case of your fishes, from heart to body through gills. Only 2 chambers is there. Since there are only 2 chambers here, from heart deoxygenated blood to gills. In gills they breathe. Fishes breathe through gills. Fishes is breathing through gills, right? So it's getting oxygenated to the body. Body is consuming the oxygen. So deoxygenating back to the heart. So heart is just, not playing an important role here. It is just transferring directly. There is complete mixing of blood. In the case of your single circulation. Single circulation, there is only one circulation. Single means what? Only one. Single means what? One. So one circulation is going on here. Clear? This is your single circulation. This is your single circulation. Clear? Clear, students? This is single circulation. Single circulation is happening. Only one. Now, in the case of your double, incomplete double. In the case of incomplete double circulation, this happens in your amphibians and reptiles except crocodile. Except crocodile, this happens in the case of your amphibians and reptiles. Now, in the case of your amphibians and reptiles, 3-chamber heart. They have a 3-chamber heart. It means heart has 3 chambers. That is left atrium, right atrium, and a single ventricle. Ventricle is single here. A single ventricle where mixing of blood happens here. Again, mixing of blood happens. Again, mixing of blood happens. So here, from your gills, lungs, what is happening? oxygenated blood is coming to your left atrium, oxygenated blood is getting mixed here, then it is getting oxygenated blood is transferring to your where, or from your body it's transferring to your body, oxygenated blood is transferring to your body from here. Can you see from your gills? They're breathing through gills, they are breathing to gills. When breathing is happening, oxygen blood is coming, oxygen blood is sent to where? To different tissues, different body is using the oxygen. When body is using the oxygen, it is going back to what? It is going back to your right atrium, and where the blood is again mixing here, and then once it gets mixed here, it is being sent to your again to your gills. Again it is see, this is a type of double circulation. That is, gills is involved, chambers are involved, body parts are involved, but there is mixing of blood is happening here. In this case, mixing of blood is happening here. That is, gills is breathing oxygenated blood here, is going to where? It's going to different tissues. Tissues is consuming the oxygen. Once oxygen is consumed, it is going back to your right atrium, where it is getting mixing, blood is mixing here again. Then it is going back to the where? Gills, and again gills and lungs. Again, that is deoxygenated blood is going here. This is also deoxygenated blood. Then it is going to gills. Again, it is getting oxygenated and it is coming back. Very simple, very simple, very easy. This is incomplete. Why incomplete? Because there is mixing of blood is happening. That's why it's incomplete. Clear, students? Anyone any doubt? Anyone any doubt here? Students, anyone any doubt? Anyone any doubt here? Anyone any doubt? Hello Vishal, welcome back. Anyone any doubt here? Students, any doubt? This is very simple. The only thing you need to understand is double circulation. Any doubt? Any doubt, Bala? Any doubt, Varshini? Any doubt? Sir, which organ transplant is most difficult in humans? I think it has to be brain. It has to be brain, which is not possible. Which is not possible. Brain transplant, again, very difficult, not possible. I think it's going to be, if it's going to be difficult in the future, it's going to be brain only. But for now, I think it's heart only, because heart you have to keep outside, again it has to work. Right? Now, in the case of humans, or in the case of your birds or mammals, or in the case of certain crocodiles, right? In the case of crocodiles, gills, lungs and skin, we breathe through lungs. We breathing through lungs. Gills in the case of your certain fishes, skin in the case of certain amphibians. What is happening? Blood is entering into your, wait, blood is entering through the left atrium. I've drew the box. Remember the box. Blood is entering into the left atrium. That is oxygenated blood. When oxygenated blood is sending, see here, it is not transferred from here to here. Can you see here? From left atrium, blood is transferred to the left ventricle. From left atrium, blood is transferred to the left ventricle. From left ventricle, the blood is transferred. Left atrium to left ventricle is getting transferred, and then it is sent to your other body tissues. Again, from other body tissues, deoxygenated blood is sent through superior vena cava. In case of humans, superior vena cava is taking inside. That is, blood is going to your right atrium here now. From here, right atrium is directly going to here. No, from right atrium it is going to the, right atrium going to right ventricle. From right ventricle here, now it is going to the pulmonary veins. Remember, pulmonary artery was coming here. Lungs, it is going back to the lungs. Amorphous means this not clear, right? Amorphous is also powder. Crystal opposite is amorphous, right? Crystal opposite is amorphous. Crystal is like completely clear, amorphous is not clear, right? Next question, that is double circulation. Here, this is your complete double circulation. What we have been learning is double circulation. This is what we have been learning, the complete double circulation here from lungs to tissue. Oxygenated blood, deoxygenated blood is transferring. Pulmonary circulation, systemic circulation. Oh, there has to be a question here. It's missing. Never mind. Now, I think someone was telling this. Someone was asking me this in the starting. Why does the liver receive blood from two different sources? Someone was asking this question back then. Why does the liver receive blood from two different sources? The answer is what? It's because of hepatic portal system. That is a unique vascular or circulatory connection between the digestive tract and the liver is called as hepatic portal system. So someone was telling the blood is also from your liver. Here it is the liver. It is from your digestive tract and also from the liver is called as hepatic portal system. Now one more thing. The heart pumps blood, but does it also need blood? Simple question. Who can answer this question? Who can answer this question? Vishal, I would advise you to ignore anyone who is not studying in the chat. Anyone who is not studying in the chat, please ignore them. The heart pumps blood, but heart needs blood. Does the heart need blood? Yes, it needs blood. That is, coronary arteries supply oxygenated blood to the heart muscles. Coronary arteries, coronary veins drain away the deoxygenated blood from the heart muscles. Yes, it needs. Yes, it needs. That is coronary artery and cardiac veins. Skip that. Let's understand heartbeat now. Can we start with the rhythm? Can we start with the rhythm? How is the blood pumped? Cardiac rhythm. Yes. It also needs blood. Heart also needs blood. Can we start with the lub-dub? Can we start with the rhythm? The cycle, what exactly happens? This is where it's very interesting. This is where so many students go wrong here. Amazing. Cute pretty Caroline is also here. Oh. Let's start. So basically there are multiple stages. When the heart pumping, in the movement of the blood, there are multiple stages. Listen very carefully now. 100%. Okay. When the blood is moving, there are multiple steps. The first stage is early joint diastole stage. What is the meaning of diastole? Diastole means relaxed. Remember this now. All of you, please remember this very carefully in your mind. Diastole means what? Relaxation. Systole means contraction. Contraction means systole. Diastole means relaxation. Right? Relaxation. So here what exactly happens? What is the contraction is happening? Which ventricle is contracting? Which is relaxing? Let's understand. The first one is your early joint diastole. Meaning of diastole? What is the meaning of diastole? Diastole means relaxed. So early diastole. In early diastole, everyone is relaxed. Everyone is chilling in the house. That is your ventricle, diastole, relaxed. Artery, your atria is also relaxed. Everything is relaxed. Everything is relaxing. What about your bicuspid and tricuspid walls? That is your atrioventricular valves are also closed. Semilunar valves are also closed. Everything is closed. Nothing is open. What happens to the blood? Blood enters the atria through veins. So blood is entering now. Blood is still entering. But can blood go into the ventricles? No, it cannot. But when can it go? That is late joint. That is, first one was your early. The second one is your late. That is late joint diastole. In late joint diastole, still everyone is relaxed. Still everyone is relax. That is your diastole. That is ventricle is also diastole, and your artery is also diastole. That is both are relaxed. But your atrioventrical valves, that is what? The bicuspid and tricuspid valves open up. When bicuspid valves and tricuspid valves open up, what happens to the blood? Blood is sent. Blood is sent. Right? The blood is sent to your ventricles. That is what happens now. AV valves are open. Now AV valves are open. That is in your late joint, diastole. Everyone is relaxed. But these valves are opening, right? I told you. Atrioventrical valves are opening. When valves open, blood is filling now. That is, some blood enters the ventricles from your arteria. So from your artery, the blood is getting transferred to your ventricles. So blood is transferring. How much blood? 70% of the ventricles will fill up here now. 70% will fill up. Done. First stage is done. First stage is done. First stage 1 is done. Now second stage. Now before second stage starts, what exactly happens is, there is something called as signaling in your heart. That is, a signal goes. A signal is released from your SA node. SA node releases a signal. SA node releases a signal. And because of this signal, right, because of this signal, so once again, let me say the question. So whatever the artery is in the systole, that time ventricle is in distole. Are you talking about which stage? Varshini, if you're talking about your first stage, in the first stage, both are in relaxed. Both are relaxed. There is no contraction. Whenever there is relaxed, there is blood. What is the question? I don't understand you. Whenever arterial is in systole, that time ventricle is in diastole. No. It is not always like that. It is not always like that. Your version you're asking the doubt, that will come in your stage 2 and stage 3. I'll get there. See here, stage 1 relaxed. In stage late joint, this will open. When this opens, both your bicuspid and tricuspid open up. Blood is being filled now. How much blood? 70% of the blood is filling up. When 70% of the blood is filled up here now. Now in second stage, what happens? In second stage, that is stage 2, that is atrial systole. Your atria is undergoing systole. What is the meaning of systole? Meaning of systole is contraction. See, that is your atria is getting systole. So your atria is systole, but your ventricle is diastole. I'll tell you why. Listen carefully now. Now if only your atria is getting systoled, this and this atria, left atria, right atria is getting systoled. Why? Because of the signal from your SA node. If this is getting systoled, if it is in contract, imagine you have a bottle here. I have this bottle has 30% liquid, 30% blood. Now what if I squeeze the bottle? Rest of the blood will also come out. Similarly, when it was relaxed, some blood went. How much? 70% of blood went. Now you are what? Now you are spamming it. So what do I say? When you are like crushing it, when it is contracting, that is your atria is getting contract. When you contract it, rest of the blood will flow down to the ventricle. But what is ventricle? Ventricle is still chilling. That is, ventricle is relaxed. Ventricle is in diastole. But atria is like, pressing. Right? Atria is like, getting crushed. So all the blood transfers to the ventricle. Rest of the blood pumped from the atria to the ventricle. So blood is getting pumped to the ventricle. That is your stage 2 because of the SA node signaling. Now third stage. Now what exactly happens in third stage? Third stage, what happens? Listen very carefully. In third stage, your AV node. In the third stage, the AV node sends signals now. Once AV node or your bundle of his sends a signal, what happens now? In the third stage, that is ventricle systole. Now your ventricles are undergoing systole. Ventricles are like systoling. They're crushing now. When ventricles are crushing, what exactly happens? Now one more thing which you need to open here, see here is, remember in the second stage, this valves opened up. The blood is coming here. But what about these walls? These valves are closed. These valves are closed. That is, semilunar valves are closed. But in the stage 3, when your ventricles are like, getting crushed, when ventricles getting crushed, when they are contracting, what happens? This one here, bicuspid and tricuspid walls close up. Why? To make sure the backflow is not happening. To make sure the backflow is not happening, they are getting closed up. Closed. Now, what about your atria? Atria is now relaxed. Its work is done. All the blood has been poured down. Work is done. But what about your ventricles? Ventricles are contracting. And what happens to the blood? Ventricles contract, cause the AV node valves to close. The AV valves are closing now. So where is the blood now? Blood is in the ventricles. Blood is in the ventricles now. What about semilunar valves? They are still closed. But in the case of your late, this is early ventricular. In late ventricular, what happens? In late ventricular, your ventricles is still systoling. They're still contracting. Atria is still relaxing. But what is happening to your atrial wall? This is closed. This was closed. But what happens to your semilunar valves? Your semilunar valves will open up now. When semilunar valves open up and you're crushing the ventricles, if you're still crushing the ventricles, the blood is now transferring to the what? Aorta and pulmonary arteries. That is, ventricles contract, causing the semilunar valves to open. Semilunar valves are opening, blood is pumped into the arteries. Blood is pumped into arteries. That is the complete rhythmic movement. Is any doubt here, anyone? 4 stages, I told you, 3 stages. 3 stages. First stage has 2 parts, last stage has 2 parts. Middle we have your early ventricle systole. You have atrial systole. In atrial systole, atria is getting crushed. If atria is getting crushed, blood is sent. And in the case of your third stage, ventricles getting crushed. In early stage, this is getting closed. But in the late stage, this valves are opening up and blood is getting transferred. Is this clear, students? Any doubt here? This has to be clear. See, this diagram makes it very clear. Diagram will make it very clear. Any doubt here? This is how exactly the blood flow happens in your body. First, I'll repeat again if you want. First stage is your first step is your early joint diastole. Everyone is relaxed. All the valves are closed. Everyone is relaxed. No blood flow is happening. In the late, what happens in the late? These valves are opening up. Once this valve, so it is bicuspid and tricuspid valves open up, all the blood is flowing into your ventricles. But in the late joint, now what happens? A signal from SA node is coming. Once the signal is coming, what happens? Atria is getting crushed. That is undergoing atrial systole is happening. Once they get crushed, rest of the blood is transferred to your ventricles. When you're getting crushing it, 30% is also going, right? And these valves are open still. Why? The blood needs to go. But in the case of your late ventricle, when early ventricle systole, when they're getting crushed, what happened? These bicuspid and tricuspid are getting closed. Blood is not flowing back. But in the case of late, what happens? Your semilunar valves open up and blood is sent to your aorta and pulmonary arteries. Pulmonary arteries. Amazing. What causes sound? Very important. Due to the closing of valves in the heart, that is bicuspid valves, tricuspid valves closing and opening, the semilunar valves closing and opening, that is due to closing of valves in the heart, the sound is coming. During each cardiac cycle, 2 sounds are produced. The closing of atrioventral valve, that is bicuspid and tricuspid, first sound, lub. The closing of semilunar valves in the late stage, in the late ventricular systole time, that is your second sound, that is dub. Lub, dub. That is your cardiac cycle has joint systole, atrial systole, and then you have joint diastole, atrial systole where atria is getting compressed, and ventricle systole where ventricles getting crushed. 3 stages. In this we have early stage, late stage, early stage, late stage. Clear? Amazing. So who can answer this? Who can answer this? What is the duration of one cardiac cycle? Students. Any doubt here? This, what I just explained, right? This concept here. This concept is most confusing to students. This concept is the most confusing to so many students who are still not able to understand how is the blood moving, what are the different stages, what is the meaning of, you know, systole, what is the meaning of your diastole. Duration of one cardiac cycle is 0.8 seconds. Heart beats 75 in 1 minute. So 72 points. That is, one cardiac cycle is 0.8 seconds. Amazing. All of you are able to answer. 70 times. So how much blood is pumped per ventricle? Per ventricle during each cardiac cycle? That is nothing but each ventricle pumps approximately 70 ml of blood. That is called as stroke volume. So what is the meaning of stroke volume? Amount of blood pumped by each ventricle. So each ventricle pumps about 70 ml. That is called a stroke volume. Now how much blood is pumped in 1 minute? Now how much blood is pumped in 1 minute? Who can tell me? How much blood is pumped in 1 minute? How much blood is pumped in 1 minute? That is heart rate into stroke volume. 70 to 72, that is approximately 5 liter per minute. 5 liter of blood is pumped per minute. 5 liter of blood is pumped per minute. Can be defined as volume of blood pumped out by each ventricle per minute is 5 liter. Electrocardiogram, ECG. Now let's understand ECG now. Graphic record of electrical current produced by the heart. Heart is producing electric currents. SA node, AV node, bundle of His, electric currents are getting produced. Right? All this you have to take into account now. Now how does the heart produce electric currents? That is your SA node, AV node, bundle of His and Purkinje fibers. I know I should have made this in the starting. This was supposed to come in starting, but it's coming now. That is, SA node is present in the here. This is your AV node, atrioventricle node. Then you have your bundle of His. Purkinje fibers are present towards your ventricles. What is SA node? SA node is also known as synovial arterial node, found in the region of right atrium. Here, top right corner, you have your right atrium. Generally generates action potential. What is the function of your SA node, AV node? Action potential is generated. Causes the atria to contract. I told you, atria is contracting. In the process, atria is contracting to release that complete blood because of action potential generated by your SA node. Conducts the action potential to the AV node. What is AV node? Atrioventricle node. Passes the action potential to the bundle of His. It is like your running race. Varshini has a doubt. This is like your relay. You pass the baton in your relay, right? One after another, it goes away. Similarly, from SA node to AV node to bundle of His. Bundle of His action potential is then helps in contraction of the ventricles. I told you, second signaling, bundle of His, ventricle contracts. Ventricle is getting contracted now. Why? Because of bundle of His and AV node. Here, I told you the steps. The third stage is what? Ventricles are getting contracted because of the signals from bundle of His and AV node. Then nodal tissues regulate the beating of that is the heart is called as myogenic. That is, hearts keep on beating is called myogenic. Now next concept, the last concept before the heart disease is electrocardiograph or electrocardiograph. The father of electrocardiograph is your Einthoven. So this you also have different term. Bombay blood grouping, don't worry, it's very easy. I can explain this in one shot. I'll make a YT shot on it and I'll upload it on my this thing Telegram channel and you can watch it there. Bombay blood grouping, nothing to worry. You don't have to watch an entire movie. 1 minute you can explain, you can understand it. Okay. Yes, I can drink some water. So while I drink some water, I want everyone to like the video. Oops. While I drink some video, I want everyone to like the video. I can see only 15 likes or 14 likes. I can only see 14 likes on this video. I want everyone to like the video. Come on. So see, the main part is over. If you understand the functioning of SA node, if you understand the function of AV node, what is the contraction, what is the diastole, electrocardiogram is the most easy part. Okay, it's time to, I think I have to block them. I think it's getting annoying, right? Oh, my legs. Done. At this point, this was getting annoying. I had the patience of an elephant, but I'm like, it's fine at this point. Inge, Rendu, Peru, YouTube as messenger. You're telling them they're using YouTube as a messaging platform, right? Bala, you're trying to tell that they're using YouTube as a messaging platform. I had a patience, but now it's gone. I waited all this while. I waited all this while for them to stop, but they were not ready to stop. Anyway. Anyway, let's start with electrocardiogram. Electrocardiography. So what exactly is P? What exactly is your QRS complex? What is T? And why are we having this gap? Why are we having this gap? What is the P? Let's understand each and everything. Let's understand each and everything. Ready? When did we start the class today? We started the class at 7:40, right? 7:40 to 8:40 to 9:40 to 10:40. Is it 10:40 yet? 7:30. But I think 3 hours will be over. Next 20 minutes, the class will be done. All the diseases, everything would have been. I can understand Tamil. I can understand Tamil, obviously. Obviously, I can understand. I'm from Bangalore. I can understand Tamil, Telugu, Malayalam. What do you think? Chemistry. So we know sodium is reacts with oxygen and water, right? So don't worry, Carolyn. Even I have done it in my college days. So I took sodium, I cut it with a knife. Actually, I cut it with a small, what do I say, normal knife, and I put it in water. There's no burst and all. There was no proper burst, but there was small, some effervescence kind of thing came. I didn't see a burst and all. This is nothing. One of my classmates bought pepper spray to the class and she sprayed pepper spray, and the whole class was crying. Now, so you understand electrocardiogram. Now, what is the P wave here? What is the P wave? This part, first part, that is small upward wave here. Small upward wave is your P wave. That is when arterial contraction is happening. When arterial contraction, the first one, remember, when arteries contract because of your what? SA node. When artery is contracting because of SA node, we get the P1, small bump we get. So remember, we put electrodes here. One both on your wrist and on your left leg. They put one conductors through which they measure the electrical signals. Sometimes even on the chest also. Okay, because of that they are able to measure all the signals from the earth. So signals from your body. That is small upward wave here. This one, that is atria is getting contracted. Remember I told you that is depolarization is happening. And why is happening? That is activation of SA node. I told you when SA node gets activated, signals of contraction spread throughout the atria. When SA node is getting activated, the artery is getting contracted. When artery is contracting before the signal, they're trapping the signal. Remember in your electrocardiogram, the signals are getting trapped. So when the signal is going from here, right, from your SA node, that is your P. That is the P. Now what is your, the small gap here, that is your short straight line. Short straight line is AV node delay. So before AV node, the signal gets to AV node, the small delay is there, right? The small delay is your AV node delay. Then you have your QRS complex, that is large upward triangle, small downward deflection, and ending, that is downward. This is a result of what? This is a result of impulse from AV node travels through the bundle of S. When the signal is traveling from here, see, can you see, this is your AV node. From AV node, the signals are transferred into your bundle of S. AV node from bundle of S. That spread of impulse to the ventricles. When the spread of wave goes to the ventricles, the signal is going. Now you're trapping that signal. When you trap that signal, that is your ventricle contraction is happening. The stage 3 ventricle contraction from bundle of S, because of bundle of S and your AV node. That is your QRS complex. That is the QRS complex, because of your signals from your bundle of His, signals from your AV node is being trapped. Why 3 electrical reeds? Because we have 3 places in your body which produces signals. Here, one on your wrist, here on 2 wrists, one on your left leg. Next one, you have a small line here again, that is upward dome shaped wave. Here, T wave, that is your ventricle contraction repolarization. Ventricle contraction late stage. In the late stage is happening, that one. That is the small bump here. Now when you end here, right, the end of this T wave is nothing but, end of T wave is the complete ventricle is undergoing diastole. It is relaxing again, right? That relaxing is this one here. The relaxing is this one. So when the T wave comes, right, when the T wave is there, once you see the T wave, the one cardiac cycle is over, your ventricle is now relaxing. Ventricle is relaxing. This person is coming in. How do I block? I have to block these people. Done. Ouch. So no MCQs today because the concept was so big. If I add MCQs today, right, if I were to add MCQs, this session has become at least 11 o'clock. No MCQs today. Oh yeah, no MCQs today. What is significance of EVG? ECG can determine the heart rate of an individual. That is, deviation from this shape at times indicates the abnormality in a disease. So a doctor will listen to this and you can always tell the heart rate. Now what is blood pressure? Oh, finally we come to the diseases. Let's start the diseases. Let's start the disease. One second students. Let's start the diseases. Let's start the final stretch of this chapter, that is the diseases. Once we do the diseases, right, once we do the diseases, everything will be over. Ready? Yaru Samay Nega means I don't know that one. Can I start the last part? Diseases. Questions have always come from diseases. If you know the question paper pattern on this chapter, the questions always come from diseases. You can find at least one question from this chapter, one will be there in diseases. Last part, let's finish it up. This is a big chapter. Trust me, we want to finish the entire physiology in 2 weeks. This week 4 chapters will be done. Okay. Parvati is also saying, Parvati, welcome to the class. Parvati is saying, yes sir, we are ready. Hepatic portal system, sir. I told you hepatic portal system, nothing to explain there. Hepatic portal system is mainly because of your livers. From liver is, your intestine and every liver is getting two different types of. There's nothing to learn there. If you want me to talk about hepatic portal system, I'll bring a numerical on this. I'll bring a question on this, and I will discuss it. Voila. On my PYQ series, I'll bring a separate question from hepatic portal system, and then explain the hepatic portal system with the help of a question there. Okay. Now, what is blood pressure? That is force exerted by blood against the walls. See here, this is your walls. So blood pressure is always going towards the walls. So if there are walls are getting what? A lot of blood. Right, the walls are getting a lot of blood pressure here. Can you see? Blood pressures exerted by the blood. That is a drive flow force exerted by blood against the walls of the blood vessels. If the blood vessels get against the walls, if you put pressure, that is your blood pressure. Drives flow blood from your heart through the arteries. So increase in blood pressure is very bad for your health, because this can be measured. This is the measurement, not your smartwatch. This is a measurement. That is graphenometer is your device used to measure the blood. That is normal level is 120 and 80. Now what is 120 and 80? 120 and 80. That is your systole and diastole. That is contraction, relaxation. Your artery contraction and your ventricle contraction. That is the 40 and 120 mm 80. That is blood pressure is also called as your hypertension. That is also called as high blood pressure. If your systolic pressure, I told you, systolic and diastolic. When the systolic goes above 140, what is systolic? Systolic means your compression. During compression, if the blood pressure is going more than 140, it is very bad. If you relaxed state, when it is relaxed stage, if there is relaxed stage, it should be less than, it should be almost 190 mmHg. What are the harms? Heart, brain, kidney, eyes can also be done. What are the causes? Increased cholesterol level, stress, smoking, and kidney disorders can lead to hypertension. Next one is your coronary artery disease, that is CAD disease, that is coronary artery disease, also known as atherosclerosis. Atherosclerosis, very different. There is a difference between atherosclerosis and anteriosclerosis. I'll tell that. Can cause brain damage, brain hemorrhage, yes. Also known as atherosclerosis, caused by the building up of the plaque. Cholesterol is adding up. That is calcium, fat, and fibrous tissue makes lumen of arteries narrow. If the lumen is becoming narrow, the blood is not flowing properly. Affects the supply of the blood to the heart muscles. Supply of the heart is because of the accumulation of fat. Next one is your angina. Angina is sudden pain in the chest. The sudden pain in the chest is your angina, also called as angina pectoris. That is not enough oxygen reaching to your heart muscles. A sudden pain in the chest will happen, right? It happens between the 20 to 30 people. 20 to 30 age group, it actually happens. Symptoms: acute chest pain occur in men and women. The last one is your heart attack. What is also called as myocardial infarction. Plaque ruptures and causes blockage. The plaque will rupture and cause the blockage and damage or death to the cardiac muscle tissue. If the damage to the cardiac muscle tissue happens, heart attack happens. Blockage or dead muscle tissue. Complete blockage will happen here. See here, your CAD, plaque formation, angina, hardening of the blood to get enough through, that is sudden acute chest pain. Heart attack is there. Plaque cracks the blood clot and blocks the entry. So whenever this blood clotting happens, if blood clotting, coagulation, that is clotting of blood happens in the heart, very bad, very bad. Why, Vishal? Why do you need my age? What is heart failure? Heart is failing. This is not the breakup thing. This is the different thing I'm talking about. It is heart does not pump enough of the blood. If heart is not working properly, if the heart is not able to work, it's not able to do its function properly, that is heart failure. That is also called as congestive heart failure. Congestion in the lungs. Why is this not going ahead? Okay, the slides have been stuck. Amazing. And the presentation has closed. Presentation has been closed. I had one more slide to show you. I don't know why that slide is not coming up. One second, let me just bring it back. Where is the slides? Downloads. I've talked about one difference is there. I want to show you the difference. Where is the difference? Here, no, this is still there. No, we can't go there yet. There's one more 2 or 3 concepts which I have to teach you, that's all, and we'll get to that. Yes, it's there. Board failure. Regulation of cardiac activity, it's there. Don't worry. Now, what is cardiac arrest? That is, heart stops beating, the blood flow, that is to the brain and the vital organs, the heart, everything stops. That is your cardiac arrest. Now, what is RTO? That is, what is happening? Now, what is arteriosclerosis? That is your hardening and loss of elasticity of arteries. So, if arteries have become loose elasticities. So, for arteries, what happens when the arteries pumping the blood, when the blood is passing, they need to be very flexible. If the flexibility is gone, right, that is called as your arteriosclerosis due to the calcification of plaques, walls and ruptures due to the loss of elasticity. If the elasticity is gone, if the elasticity is completely being gone, what happens? They become, your arteries become very tight. That is your arteriosclerosis. So, know the difference between arteriosclerosis and arteriosclerosis. In atherosclerosis, your formation of plaque happens. In arteriosclerosis, thickening of the inner walls of the central wall of the arteries because of the loss of elasticity has been lost here completely. Elasticity has been completely lost here. The last one is the regulation. The last one is your regulation of your cardiac activity, that is your, because of your brain, that found in your lower part of your brain, that is central cardiac function, that is your cerebral part of your, that is your cerebrum. It has this control over these 2 nerves. I'll tell you. 2 nerves are there. Very simple. This is your vagus nerve and sympathetic cardiac nerve. That is, 2 nerves connect the medulla region of the heart. The 2 nerves connected to the medulla region of the heart is sympathetic and parasympathetic. Parasympathetic is your vagus nerve. Sympathetic is your sympathetic cardiovascular nerve. 2 nerves which make sure that heartbeat will go faster, heartbeat will go slower. That is your sympathetic increases the rate of heartbeat. Sympathetic will increase the rate of heartbeat. That is, strengthens the ventricular contraction and thereby cardiac output. Sympathetic means what? Sympathetic will increase the heartbeat. Parasympathetic will decrease the heartbeat. Parasympathetic decrease the rate of heartbeat, speed of conduction of action of potential is decreased completely. The other one is your hormones, such as your cardiac output. Adrenal gland will also increase the heartbeat. Adrenal gland will increase the heartbeat completely. That is fight or flight. During fight or flight, because of the hormones, the heart beats very fast. When you're scared, heart is beating very fast. When you're doing something wrong, heart is beating very fast, right? That's all. That's the end of it, I guess. That's the end of it. That's the basic last part is your, not that important. The main important part here was your cardiac rhythm, the heart, how the functioning of the heart, what is ECG, what is each point in ECG, and then what is the different type of cells, different type of blood, what are the different components of blood. Okay, those were all the most important parts. Everything has been covered. Trust me, nothing out of this will be asked in your examination. Nothing also this will be asked. If anything is asked, I will make sure I'll cover it also. Okay, nothing out of this will be asked. I hope this session was very helpful to all of you. If this session was very helpful to you, I want everyone to like the video and subscribe to the channel. And I am very, very hungry. I am very hungry, man. Last time I had lunch was 1. I think I should eat the lunch later when I have class till late classes, right? I think I should eat a lunch very late. Right? So thank you so much. Any doubt here, ask me. No. One is the hepatic nervous system. I will make a video on it separate when I take questions, and I take some questions, right? PYQ series. You know, right, once I teach a session, we take a PYQ on it. So during the PYQ session, I'll discuss it there also one more time. Every question will be dealt. Do not worry, we'll make sure you learn every question from this chapter. Okay, that'll be done. And also Bombay blood grouping, I'm planning to make a video on it. Okay, so good night everyone. Thank you so much for joining. Thank you so much for joining today's class, and oh god, I have to eat something. So then, bye bye students. Have an amazing night. Sleep well and take care students. Bye bye.